sir.
Thank you.
Good morning, ladies and gentlemen. Welcome back to the day 2 of the Indo-Malaysian Health Science and Technology Summit 2025. I hope you had a refreshing evening yesterday and are ready for another day of learning, innovation, and collaboration. We begin with scientific session. Our first speaker of this morning is Dr. Mahendran Cheah, Associate Professor, School of Pharmacy, Monash University, Bandar Sunway, Malaysia, joining us today in online mode. Sir has established himself as a leading contributor to both teaching and research excellence. Sir primary research interests lie in computer-aided drug design, synthetic medicinal chemistry, molecular docking, and QSAR studies with a particular focus on therapeutic areas such as cancer, diabetes, and neurodegenerative disorders. His expertise in combining computational and experimental methods has resulted in impactful contribution to the field. He has published over 40 peer-reviewed research articles in reputed international journals and authored several book chapters. His work has been widely cited, reflecting the relevance of his contribution to pharmaceutical sciences. In addition, he has successfully guided postgraduate and doctoral students, nurturing the next generation of researchers. Sir is also the recipient of several prestigious awards and fellowships recognizing his academic achievement and innovative research. His dedication extends beyond research as he actively participates in academic development, conferences, and professional bodies. For his topic, Sir's topic for the today talk is current and future approaches in drug discovery and development from
Yeah, hi everyone, and thank you very much to the MC for the kind introduction. I take this opportunity to thank the organizing team for inviting me to take part of this international conference. Maybe can you please allow me to share my slide? Yeah. I'm sharing my slide. Yeah, can you see my screen now? Now, okay, the PowerPoint slide. Can you see? Wait 1 minute. Now, is it okay? Now, you can see the slide. Okay. Okay, just let me continue. So, in case any interruption, just let me know so that I will stop presenting. Then maybe after that, maybe I will continue again. Okay. Anything, just let me know. Thank you. Okay, once again, yeah, thank you very much for inviting me for this international conference. The topic I was selected for today's presentation is current and future approaches in drug discovery and development from natural products. Why particularly I chosen this topic is because, as you know, in pharmacy discipline or pharmaceutical science discipline, we have various departments like pharmaceutical chemistry, pharmacognosy, phytochemistry, pharmacology, pharmaceutics. There are many departments, right? So in that department, the primary source of their research is natural products. Since we know the natural products are safe and more efficacy when compared to the synthetic molecules which are currently available in the market. So that is the reason most of the researchers, their primary source is natural products. And natural products means, I think most of us, I think, might be thinking that is only obtained from the plant source. So that is wrong actually. So natural products are also obtained from other natural source, including plants and also animal source as well. So plants, as you know, there are plenty of drug molecules which are currently available in the market. Similarly, I think you know insulin, which are obtained from the animal pancreas, and also penicillin obtained from microorganisms. So there are many other natural sources, including the mineral source also. There are some minerals which are used in the form of drug candidate in the market. But discovering a natural product or natural drug candidate is not a easy task. It is a tedious process which starts from extraction, isolation, then characterization, followed by we need to do some in vitro or in vivo studies, all preclinical studies. Then only we move on to the clinical studies. So it is a long process, and it takes minimum 10 to 15 years of time to bring one drug candidate into the market. So due to that reason, I think many researchers, I think some of the researchers, even they have been chosen small molecule, which mainly from the synthetic molecule, and they do some derivatives and they go for in preclinical and clinical studies. Even though some they choose as a synthetic molecules as their primary source due to tedious process in natural product research, still there are plenty of compounds which are available from natural source that can be used for the treatment of many emerging disorders. And as you know, the natural isolated compound still remains as a forefront of modern therapies for cancer, infections, inflammation, and some metabolic disorders as well. And as you know, our researchers they put lot of effort to bring a new candidate to treat against some disorders, and also institutions. There are many institutions, not only in India, maybe in overseas as well. There are many institutions and also government agencies. They usually give lot of funds for especially the project which are related to natural products. And this particular slide, I'm going to show the global view of the role of pharmacy in drug discovery and development. So here, as you know, any drug discovery process, which always starts from target identification and validation of against disorders. So this we can predict the biomarkers, then we do the validation and identification. So this all can be done with the omics technology, where we supposed to use metabolomics, proteomics, all omics technology to predict the biomarkers. So once we predicted the biomarkers, then we go on to the lead identification. Once we done the lead identification, then we do some modification to increase the efficacy of the molecule and also to reduce the toxicity of the particular lead compound. Then what we do usually, we go for in vitro studies by using cell lines or maybe some chemical methods to find the effectiveness of the particular lead molecule. Then once we found it is biologically active, then we move on to the pharmacological studies, where we use animal models, that is in vivo studies. And also at the same time, we also perform the toxicity profile. Then we move on to the clinical trials, where phase 1 is done with the healthy volunteers to check the safety of the molecule. Then phase 2 with the small number of maybe diseased patients to check the effectiveness of the drug. Then finally, we move on to the phase 3 clinical trials, where we conduct with the large number of patients. So this is the process of drug discovery and development, either from natural source or maybe from the synthetic molecules. So this particular process, it takes minimum 10 to 15 years of time to bring one drug candidate into the retail pharmacy or maybe into the market. And as you know, in pharmacy discipline, you know, as I mentioned, we have various discipline, all the disciplines, all the departments, everything is interrelated to the drug discovery and development process. So as a pharmacist, we must know our role, even though we people are maybe one from maybe chemistry, maybe other from pharmacology or biotechnology, but everyone play a major role in the drug discovery and development process. So here, as I mentioned, my background is natural product research. So here in our laboratory, we do isolation, characterization, then followed by we do the structural modification of natural compounds. So here in this particular slide, I'm going to show what is a traditional research. Since my topic is what is the current research and how we can move forward to avoid all the challenges that we face in the drug discovery and development of natural products. So as you know, any natural product researchers, it always starts from the identification of plant material. So how they select or identify the plant material? So based on the traditional uses, right? So once we come to know the what is the traditional use of that particular plant, or maybe we do the literature review. Maybe from India, I think you all might be using Indian medicinal plants, Materia Medica, to find what is the traditional use or ethnomedical use of that particular plant. So then you do some literature review. After the literature review, then we go for the collection of medicinal plants. So once we done the collection, we do the authentication with the help of botanist. Then we move on to the extraction process. So extraction process, I think most of our researchers, they usually use polar solvents, because as we know, the secondary metabolites, especially the polar compounds, are biologically active. So we use polar solvents to isolate all the secondary metabolites. But in our lab, what we use is we use the mixture of solvents like dichloromethane and methanol, or we use chloroform and methanol or ethanol mixing together, so that we can isolate all the phytoconstituents which are present in the plant material. Because our aim is not only to isolate the secondary metabolites, because as we know, there are some primary metabolites which are also responsible for the biological activity, and that also available as a drug candidate in the market. Then once we done the extraction, then we go for the isolation. So isolation, we usually use column chromatographic method. So where we have to pack the plant extract into the column, then we do the manual method. This is what I did during my PhD time, that is 10 years, 10 to 15 years before. But now also there are many researchers, those are doing natural product research, they're still continuing the same process. Because each and every extract, if you pack in a column chromatography, and if you want to isolate the phytoconstituents which are present in that particular extract, it takes minimum 3 to 4 months of time to complete one extract. For example, as a PhD student, if they select one extract, three extract for example in their PhD, then to complete the isolation process itself, it takes 1 year of time. And then they need to do the in vitro or in vivo pharmacological studies, then that may take another 2 years of time. So currently we have a flash column chromatography which we are using in our laboratory, where we can isolate the phytoconstituents in a faster manner, where maybe you can complete one extract within maybe 24 to 48 hours of time. So we can save a lot of time. So all the researchers, my suggestion is, those are facing the isolation, try to use flash column chromatography instead of using the normal column chromatography, that is manual method. I know there are many institutions, I think they may not be having the flash column chromatography. I know the difficulties. So what you need to do, by the time only you need to have a collaboration with university, those are having flash column chromatography. So when you approach the institution, for sure they will accept maybe your extract, and they may run for you, or maybe you can send your PhD student, or the PhD student directly go over there and continue to do the isolation process, so that they can complete the isolation within 15 days to 1 month of time, 3 to 4 extracts. Otherwise, if you do the traditional method like column chromatographic method, that usually takes 1 year of time to complete three extracts. Then after the extraction, what they usually do, they go for the characterization by using spectroscopical method, where we use IR spectroscopy, NMR, then mass spectroscopy to identify the molecular, to determine the molecular weight. Then once we done that work, then we move on to the in vitro or in vivo pharmacological studies, where we need to find the biological activity of the compound. And also sometimes the researchers, they may pass the sample to the pharmaceutical chemist, who can synthesize that particular compound and further pursue with the biological studies. So this is the traditional method of most of the natural product researchers. Even during my PhD time 15 years before, I have been followed this procedure. So while following this procedure, do you know how much time it takes for the drug discovery and development process? As I mentioned, it takes 10 to 15 years of time to develop one drug candidate to treat against disorders. So to avoid all this one, so here in this slide onwards, what I'm going to share, what are the challenges that you are facing in the traditional method and how you can overcome by using latest instruments as well as latest methods to overcome all the challenges which are facing in the natural products. As we know, there are many natural product researchers. If you ask the question what is their main challenge, is the main challenge is the quantity of the compound. Most of the researchers, so they can do isolation, they know the procedure how to isolate the compound from the plant extract, but after the isolation they may get only few milligram, like very minor quantity of that particular lead compound. So that is only enough to characterize the particular compound, means you can structural identification is sufficient, but you cannot further pursue for the in vitro or in vivo studies due to lack of quantity. So my suggestion for those are facing the quantity issues, maybe you need to collaborate with the person who can synthesize that particular compound or who is well expert in synthesizing or preparing a synthetic scheme to prepare that particular molecule. And nowadays there is a online tool also is available. There is a tool from Merck, Cynthia, where you can get the synthetic scheme. So that particular software, what can be done is, so that do the literature review, and based on the literature review, and that can automatically predict and give what may be the starting material and what are the reagents that you can use it to synthesize that particular compound. So even though if you don't get a chance to get a collaboration with the synthetic chemist or medicinal chemist, my suggestion is you can use that particular software where you can come up with a synthetic scheme and you can prepare that molecule. So once you prepare a synthetic molecule, once you prepare the molecule, then the quantity may be sufficient enough to go for the in vitro or in vivo studies. And another challenge is, as I mentioned, you can even though if you prepare a synthetic molecule, but still that may be biologically maybe not much active. So maybe you need to improve the biological activity, or maybe that may be producing some toxic effect. So how you can overcome those things? So you can overcome by using structure activity relationship studies. And before that, I would like to give some little bit information especially about the structure activity relationship. So in this particular slide, I think you can see there are two molecules, morphine and codeine. But if you look at the chemical structure, there is a only difference is this hydroxyl group is converted into methoxy group. But if you look at the pharmacological activity, morphine is 10 times more potent when compared to codeine. But structurally, if you look at, this is very minor changes in the chemical structure. So from this, I think you can understand how important the structure or functional group which are present in each and every drug molecule. Then this is one example. You look at the another example, levodopa and dextrodopa. As you all know that levodopa is mainly used for the treatment of Parkinson's disease, but dextrodopa is completely inactive against Parkinson's disorder. But if you look at the chemical structure, levodopa and dextrodopa, what is the difference? So there is no difference in the chemical structure, only their three-dimensional configuration, the stereochemistry is the shape is different. So even though the structurally it is not different, but if the shape or maybe three-dimensional configuration is different, that also play a major role in the biological activity. So why particularly I'm explaining this, even though if you have a already isolated compound and once you identify the chemical structure, you can use some method where you can produce more siblings or more similar kind of molecules, and from that maybe you can identify the lead compound as well based on some sort of technique that I'm going to cover anyway later part. So how you can move on with the structurally modified compounds. So this is one of the another method where you can produce more compounds which can mimic the natural products which may be originally obtained from the plant material. And other than that, there is one another difficulties. There are lots of procedures, as I mentioned, extraction, isolation, character. This all it takes a lot of time, 10 to 15 years of time to bring the molecule. So to avoid all this, we have sophisticated instrument, all the analytical instruments nowadays very well established where you can minimize the time, because as I mentioned it's a time consuming process. So when you use the latest instruments where the machine learning or artificial intelligence also incorporate into the some of the instruments which are available nowadays, where you can minimize the time. So all the methodologies including the extraction, even extraction, I think most of them might be choosing maceration or maybe soxylate extraction method, but there are plenty of latest method like supercritical fluid method extraction and high liquid pressure extraction method. So where you can minimize the amount of solvent and maximize um the extract quantity, where you can isolate all the phytoconstituents which are present in it. So each and every challenges there is a solution, but only thing is you must know how and what are the latest instruments are available. As I mentioned, even though those instruments are available in our laboratory, don't worry about it. Nowadays the technology is developed where you can contact the researchers who is utilizing that particular instrument, so you can get some guidance from them, and so that you can move on little bit faster, and you can reduce the time consuming process of drug discovery and development. The other important challenge is the in vitro studies or biological assay of those isolated compound or maybe semi synthetic compounds in vitro or in vivo studies. So that also you may need grant, or maybe you may need some financial support to complete the in vitro or in vivo studies. So this one also we have nowadays an alternative, which is an in silico method. I think many of the researchers, I think in India and all over the world, they are following in silico studies before they go for the in vitro or in vivo studies. Because here what you can do, you can select the proteins target against that particular disorder, and you can check based on the molecule chemical structure. You can use the software, and nowadays there are many latest softwares available like Discovery Studio or maybe scrolling the software. So that can be utilized. Even though if you don't have in our institution, contact someone who is utilizing that latest softwares, the latest version, so that you can predict the biological activity before you go into the in vitro or in vivo studies. So here in this particular software, maybe you can determine the binding efficiency of that particular molecule, and also you can compare with the standard drugs which are currently available in the market, so that at least you can predict and you can select the molecule. So in our laboratory, even we are not much utilizing the in silico studies um, for the just finding the biological activity. We are utilizing this particular in silico models to find the lead molecule, or maybe to produce more sibling molecules. Like in my laboratory, one of my PhD student is working on one of the naturally isolated compound, and she produced, I think, 200 to 300 sibling molecules, similar molecules, by using this particular software. And by using this binding efficiency, we determined and we found only 10 molecules which may be biologically active and less side effect, or maybe less toxic effect. So due to this, maybe you can minimize lot of time as a natural product researcher, because you are coming up with a naturally mimic molecule. At the same time, you already identify the lead compound before you perform the in vitro or in vivo studies, so that when you perform the in vitro or in vivo studies, that way that time you can expect the at least more, better pharmacological action when compared to the other molecules which are obtained from the natural molecules. So this is one another way. So those researchers may be doing difficulties maybe in finding the new molecule from the literature. So you can produce more sibling molecules. So you can structurally modify. And as I mentioned, the importance of structural modification, and so even very simple functional group that can give major difference in the pharmacological activity as well as in the toxic effect. And apart from that, I also would like to share one another method. Maybe you can use metabolomic studies, where you can find a large number of molecules, the metabolites, the small molecules, so which can be used for the treatment of many disorders. So this particular metabolomic studies, you can find small molecules or metabolites from the maybe biological sample, or maybe from the plant extract. So you can use LCMS, GCMS to find the lead compound as well. So this also can be used to identify the new target, mechanism of action. You can also find the safety and the efficacy of the drug, as well as you can also predict the ADMET properties like pharmacokinetic properties as well. Why this particular method I'm suggesting to the most of the researchers is, so from this maybe you can earlier you can predict the toxicity effect, because many researchers during my time, maybe 10-15 years of time before, many researchers they usually do pharmacological activities, and they come to know that is very effective against some disorders. But later when they do the toxicity studies, especially when they go for a clinical trials or maybe phase 1 clinical trials, so that particular compound failed in clinical trial because of the safety issue. To avoid that one, my suggestion to the young researchers or the faculty members, those are doing natural product research, This method, particularly metabolomics method, also you can use it to find the metabolites, as well as you can find the toxicity of that particular compound. So this way you can minimize the time of drug discovery and development process as well. And as I mentioned, there are some instruments like LCNMR and LCNMR-MS. This instrument also nowadays available. I think I'm not sure how many institution having this particular instrument, because this instrument, the specialty is you can do the isolation as well as the characterization with full of one instruments. So where the LCMS play a major role, where you can identify or maybe you can collect the fractions of compounds which are present. Then yeah, with the NMR also, which is attached with this particular instrument, where you can determine the or chemical structure or number of hydrogen carbons in that particular compound. Then you can also determine the molecular weight and metabolomic studies of this particular compound. So all together in one instrument, which is also available in some of the institutions. So this way, maybe whatever the traditional method, if you spend 1 year of time, so by using this kind of instrument, we can minimize at least 1/10 of the time. Means within 1 month you can complete whatever the study that you do in the 1 year of traditional methods. So that is what I'm here highlighting. So how, what are the challenges that you are facing in traditional methods and how to overcome in it. Okay, so my suggestion is target the major compounds. So here I mentioned some of the compounds, but I'm not working all these compounds. This all I know well existing compound, and many researchers has been already reported lot of compounds. But in our laboratory, particularly in Monash University, we have isolated compounds which are novel compound. Means, as you know, there are plenty of compounds already available in the library. It is not necessary that you need to, again you need to select a new plant, you need to isolate a new compound, then you need to do the structural modification. It's not like that. There are plenty of already compounds are available in the natural product library, which are not been fully established for the biological activities. Even during my PhD time, my even my some of my friends, they have been isolated and reported, but later they didn't further do the biological activities due to the lack of quantity. So what we need to do as a researcher, we need to identify those molecule, and we need to identify the synthetic procedure, how to synthesize that particular compound. Or maybe you can go for in silico method, where you can produce more siblings of the natural product molecule, and from that you can identify the lead compound and you can move on to the next step. So this way you can avoid a lot of incompliance, especially when you do the isolation, because as you know, extraction isolation itself, the PhD student or master student, they need to spend minimum 1 year to 1.5 years time. So to avoid this time consuming process, directly we can take the isolated compound which are already available, which are not been well established for the biological activity. And other drawbacks for the natural product is, as you know, the molecular weight of natural product is very high, maybe more than 500 or 600. And the in vivo instability is one of the major issue, because once it enters into the body, that may break down into the smaller molecules. So we cannot expect the expected biological activity or maybe the pharmacological action. And low bioavailability also one of the issue. As you know, the importance of lipophilicity and hydrophilicity, both are very important for the drug delivery. And also that's why poor solubility in water, because any drug candidate that must be soluble in water, then only it is soluble in the bloodstream, then only we can expect some pharmacological action. Similarly, in our body we have lipid barriers, where all the drug candidate must pass through. So lipophilicity also very important. So lipophilicity and hydrophilicity is very very important for the drug delivery. And also, but natural products face such issues in the drug delivery, and also problems with the target specific delivery as well. So for example, if you are working on something neurodegenerative disorder, the drug must pass through the blood brain barrier. But the natural product, the problem is because of a huge size, and also that can maybe break into a smaller particles, and it is very difficult to enter into the barriers, especially the lipid barriers, and also target drug delivery like brain or maybe some other organs that you are targeting. So my suggestion for those researchers, especially facing drug delivery kind of issues, you can try to use nanoparticle synthesis or maybe nano carriers, where you can improve the drug delivery. Also you can use self-emulsifying drug delivery methods, or maybe you can also use mucoadhesive drug delivery system. There are plenty of drug delivery systems also available apart from nanoparticles. So here I mentioned some of the non-metallic origin which can be used to convert your isolated compound into nanoparticles. And my suggestion is you can use iron oxide or maybe silica nanoparticles, which are less toxic, when compared to the other metal based non-metal based nanoparticles. And metal based nanoparticles, you can use, there are plenty of metals are available, and there are plenty of research also done by other researchers. But my suggestion is you can use silver or gold nanoparticles. And moreover, if you use silver nanoparticle, that is cost effective and also very cheap in cost, and also at the same time you can expect the nanoparticles in a better manner when compared to other metal based nanoparticles. So, in my conclusion, so as I mentioned so far, what are the traditional method that we use it for the natural product research, and what are the challenges that we are facing and how to overcome it in it, especially the time consuming process, as well as I mentioned the quantity issues. So how you can overcome with the help of medicinal chemists, or maybe there is a AI tools nowadays available like Cynthia, the Merck introduce a software which can be used to come up with a synthetic scheme of any of the molecule. So that maybe it may not 100% work out, but with that maybe you can further develop the synthetic scheme to come up with a sibling of new molecules. So even though, as I mentioned, there are plenty of researchers, they show interest on natural product research, but due to all this time consuming process, they usually taken some small molecules which are maybe synthetic molecules for their research. And also if you look at, there are plenty of new technologies are developed, and now many analytical instruments, as I mentioned, lcnms, lcnmr and ms, so where we have included all the artificial intelligence related things as well as the machine learning, where you can predict the biological means active molecule before you isolate from the plant material. And for sure in the future, even if you have one leaves for example, or maybe seeds, you can easily predict what may be the phytoconstituents. Such a technology is going to develop in the near future in 10 maybe 5 years of time for sure. So this way we can reduce or maybe minimize the time of drug discovery process, so that at least we can see products from natural products that are available in in the market to treat against many disorder. As you know, currently available drugs also, if you look at 40 to 45 percentage of the drugs which are currently available in the market are either from isolated from the natural products or maybe derived from the natural products. So still natural products have the future, and there are plenty of new novel molecules which are available in the library. So don't go beyond the medicinal plants. So select the directly the isolated compounds which are currently available in the natural product library. Select those compounds and do in silico studies, produce more siblings, more new molecules, and try to do the in silico methods where you can check the binding efficiency of the target proteins against the disorder. Then from that you can go use the software again, the Cynthia or maybe the Mix software, where you can find the synthetic procedure, because we cannot move depend all the time collecting the plant material. As you know, paclitaxel is one of the anti-cancer drug which are currently available in the market, but still we don't have the synthetic procedure. So there are plenty of opportunities are there to the natural product researchers, rather than finding a new medicinal plant again and again, or new compound again and again. So instead of that one, you select already the existing one which are not been well established, so that you are, you are minimizing the time, so that within few years of time you can come up with the drug candidate. Maybe you can move on into the clinical trials rather than doing the preclinical studies for 5 to 10 years of time. So with this I would like to thank the organizing committee for giving me an opportunity to talk in front of all the participants for this conference. This is my contact email ID and handphone number. So feel free to ask if you have any questions in related to my topic, as well as if you have any kind of challenges that you may face it in the natural products. Please feel free to contact, and we do a lot of research in related to natural product as well. And with that, once again, yeah, thank you very much, and I'm happy to answer if you have any questions.
And opportunities of drug discovery of natural products, role of medicinal chemistry in drug discovery, and in silico methods, metabolomics studies, LCNMR, LCNMR-MS used for the characterization of natural products, and I think it is very useful for the students. So thank you so much, sir, to joining us today, and everyone please give a big round of applause to the sir. Sir, I'm
Yeah, thank
finishing
you
the
very much.
meeting. Thank you so much, sir.
Thank you very much, everyone.
Now we are moving toward the second lecture of the day. It gives me immense pleasure to introduce our next distinguished speaker. We are honored today to have with us Professor Vetri Selvan Subramanian, Deputy Dean of Internalization at the Sir Jeffrey Cheah Sunway Medical School, Sunway University, Malaysia. Today, he will be sharing his insights on a highly relevant and innovative topic, smart biomaterials for targeted anti-obesity drug delivery. With obesity emerging as a global health challenge, his expertise in drug development and biomaterials promises to offer us a fresh perspective on smarter, safer, and more effective therapeutic strategies. Everyone, please join me in welcoming Professor Vetri Selvan Subramanian on stage with a big round of applause. Yes.
Good morning to all of you. My pleasure to be a part of this conference, Health Science and Technology Conference 2025. My sincere thanks to the organizing committee members and respected participants. I am Vetri Selvan Subramanian, working as Professor of Pharmacology at Faculty of Medical and Life Sciences, Sunway University. I am working as Deputy Dean of Internationalizations. I also very difficult to pronounce. When I took over the position, internationalization, is very difficult to, pronounce, but somehow I managed well. So my portfolio is mainly I'm looking for ranking of the university. Also, I'm teaching for medical pharmacology for medical students. In the past 10 years, I'm teaching only for medical students. Basically, I'm a pharmacist, but I'm teaching medical pharmacology. So with this, I'm going to give a short presentation, not for a big, long time, or very short presentations. How the biomaterials for targeted for the anti-obesity. Why I took this topic? Why obesity is important? According to ICMR, Indian Council of Medical Research, 2023-2024, they declared the next 10 years, the obesity is a major problem. Obesity is the major problem in India. Not only in India, in Asian populations. In the world also, there are many complications due to obesity. How the obesity? May be due to the genetics, maybe other factors, but main factor is lifestyle modifications, the food habits, lifestyle. So our lifestyle totally changed. Our internal gut system also changed due to lifestyle modifications. We all move to modernizations. When we updated or when we move to modern lifestyle, automatically the disease level also increasing, especially for the obese. The obesity is not only for increasing of body weight or maybe overweight. The obesity also develops several cancers, including of ovarian cancer, breast cancer, prostate cancer, also triggering of neurodevelopmental disorders, neurodegenerative disorders like Alzheimer, Parkinson, so many complications due to obese. Also insulin resistance. When the people with obese more than 10 years or 20 years is a high chance to develop the insulin resistance. So when the insulin resistance, so the diabetic mellitus, type 2 diabetic mellitus, so many complications due to obese, especially for abdominal obese. In India, more than 33% is abdominal obese. Only the abdomen only is become obese because of lifestyle modifications. So we have to take care of us. So the food, especially for fiber content food, is drastically decreased with our daily routine food. So these are the major complications. There are many aspects I am going to discuss here. How important of biomaterials, especially for obesity. Currently, we have a conventional drugs, especially orlistat. It's a lipid lowering agent, especially for weight loss. We are giving for kind of therapeutic aspects. We are giving for orlistat medicine, especially for obese patients. But even though, if the patient taking of more than 5 years, again regaining of body weights. Also, it's a developing of inflammations, joint inflammation. There are many complications with the existing drugs. So that now we are looking for novel therapy or novel finding of drugs. So in my laboratories, we are doing animal modeling because basically I am pharmacology. So we are doing animal modelings. I received the grant from Ministry of Higher Education on obesity research from Malaysian government grant. So with this, I have a postdoctoral student. I also have 4 PhD students. So they are all doing different metabolic research. One student doing for obesity, another doing for metabolic resistance, another student doing for how obesity triggering of cancer, especially for breast cancer. In India also, breast cancer is a high prevalence next to ovarian cancer. The same for in Malaysia also is a top 1 cancer is breast cancer. Then colon cancer, there are other cancers. So this is my university where I am working in Malaysia. It's a Sunway University. Before joining Sunway University, I was in Monash University, School of Medicine. So currently I am working as with a Sunway Medical School. Our university is a top 300 ranking university in the world, QS ranking. Also, Times Higher Education, we are the top 81 in the world. We have associated with several international universities, including Harvard University, Oxford Universities, MITs, and other the research intensive universities. So our universities mainly we are focusing on research, also academic, to go for quality educations and progress the quality research outcomes. So with this, our own research center located in University of Cambridge, the name of the Jeffrey Cheah Biomedical Center, Cambridge University. This is our own center located in the center of the campus in Cambridge University. So funded by Sunway University. So every year is millions of money they're sending from Malaysia to Cambridge to motivate, to encourage, to collaborate research and research activities. So with this, this is my topic is how the biomaterials very important. So just now, Dr. Mahendran spoken about the natural compounds. Definitely, the natural compounds are also very important. There are many sources of drugs, not only for synthetic source. It's a plant source, animal source, also mineral source, microorganism source. There are several sources of drugs. But majority of the drugs is a chemical source. So what is the major drawback or major gap? So when the people are taking for chemical for long period of time or chemical source of drugs, the drug itself, it may chance to develop the kidney damage. It's a kidney failure, renal, we used to call renal failure, sometimes damage of liver, because the drugs excreted by kidney only. When is every day taking of, consuming of too much chemical, is after sometimes the chemical itself to damage of is a renal system. So these are the complications. So that we are looking for which drugs may not affect the the renal functions or normal defense mechanisms. So without affecting of the normal defense mechanisms, we are looking for some novel compounds which are easily available is within the country base, like in India is a lot of traditional values. So we are looking at the novel compounds based on the plant compounds. Many of them they are telling is a plant compounds, still there is no evidence. There are many evidences. Morphine, the best example, morphine drug. Morphine is one of the is a plant derived compounds. Still we are using in modern medicine. If those people with the cancer, intolerance pain, severe pain, they cannot tolerate, immediately they give morphine drug. Also any severe accident, lorry accident or car accident or any major accident, mechanical accidents, immediately we give morphine drug. It means that the natural drugs still have a values, but when modernized, when go to the next level of advanced research. So with this, here we are looking for which compounds maybe in the future we are going to target on obesity with the biomaterials. Biomaterials mainly for bio is a living system. With the living substance, with the living system, how we can infuse, how we can fuse with the biomaterials, with the natural compounds. So here there are many pharmaceutics and chemistry experts. So how to develop the formulations? How to develop the formulations? But nowadays biomaterials also we are giving internally, but action is very local actions, not for systemic actions. So this kind of approach is very important. So before we are talking about whenever we take inside the body or internal system, we call it as is a systemic response or systemic reactions. No. Even though when we administered into the body or in the internal system, particularly act on the specific area of the locations. So this also we used to call local actions, the local responsibilities. So with this, the biomaterial mainly looking for local actions. So with this, what are the challenges, current medications? Lot of challenges, mainly affect the normal metabolic functions, including our gut system also may affected, the quality of life also affected. When the patient taking of a chronic period of time, lot of side effects. If they're taking of more than 5 years, why metformin drugs? Every 3 years they wanted to change. Because metformin itself to damage of our metabolic functions. Is a metformin itself, it may damage of our, especially for the pancreas. So that we every 3 years, why changing of the drug or some specific compounds? Because sometimes the drug itself it may cause harmful effects, toxic response. So that we are looking for novel compounds. But novel compound means everybody talking about novel compounds, but we are looking really something we need to find which is really useful for the society. Whatever we do for any lab research, we always be talking about translational approach. Everybody is a translational approach only. Whatever we do, any simple study in the laboratories, maybe the chemical test, we think that, okay, the color appearance, okay, is a pink color, oh, we found something. This is the record we observed in the lab report. No, that small findings also will helpful to go to the next level of research. With the basis of evidence, when we go to the in vitro studies, or maybe it's a next level of animal modeling, then next level of clinical trials, that is a translational approach. We cannot stop within the lab. Whatever we finding within the lab, we translate into the next level of research. So this is a translational research. We all focusing on translational approach. So we also have, personally I have 2 grants with Monash University when I was in Monash University. Still, we are continuing with the translational approach of research studies. So translational means is we have to continue with the basis of the previous studies, with the basic research, how the basis of the basic research, how influence or how give the ideas to move on to the next level of advanced research. So this is very important, but current challenge of, come to the topic, is the current challenge is lot of side effects, sometimes poor absorption of drugs, maybe the drugs cannot take is a longer period of time, maybe toxicity. So what we are looking, what innovations need without affecting of our pH, Maybe we don't want to affect our internal or systemic responses, particularly the normal defense mechanism we don't want to affect. Particularly, we going to target the proteins. So nowadays, we are targeting proteins for cancer patients. Even though National University of Singapore and Nanyang Technical University, they're focusing is a targeting of protein expressions. When too much protein expression, the severity of cancer also is a heavy. It means the severity of cancer is a highly increased. So when we are protein expression, when we stop, or maybe when we suppress the protein expressions, the level of progression also decreased. The cancer progression also decreased. There are a lot of research studies they are doing. In the part of the research studies, the future aspects, how we are going to develop biomaterials as a pharmaceutics, mainly for the drug formulation developments, how they are going to invent is a new biomaterial compounds with a bioactive molecules. So this kind of approach is really impactful. Is a recent days, before we are talking about is a nanomedicine, then now we are talking about exosomes, next level of nanomaterials, nanomedicine. Is even though is one of my students working on exosomes, is a cancer exosomes. So kind of every 10 years once, or every decades is a new advancements coming. So now we are looking for biomaterials. It's really is very impactful. Maybe it's very helpful for our communities. So with this, so basically is my background is pharmacology. So usually I'm looking for how the animal responds. So what kind of response produced from the majorly for the biochemical estimations. I am taking of blood, or we go for biochemical estimations like how affecting of liver functions, mainly for aspartate aminotransferase or alanine transferase. Sometimes we go for kidney parameters like creatinines. Also we are going for histopathological examinations. So basic studies only we are doing, but also we are doing for some kind of Western blot method, gene expressions. But this is not enough, not sufficient. We must move on to the next level of research. Then only we can, we can get or we can find the novel therapeutic approach. So with this, targeting of ligands, mainly whatever biomaterials, why we are taking of biomaterials, we target the peptides. Also we target the antibodies. Before we are talking about antibodies, what are the antibodies? Antibodies basically is a defense mechanisms. Like a lot of antibodies in our body like IgG, IgM, IgA, and the game. It's a game, you remember it's a game, IgG, IgA, G, right? GAME, IgG, IgA, IgM, IgE. But majority of inflammations mainly by the IgE, hyper-responsiveness. Any disease conditions, either is a cancer, or either is a diabetic, or either is any disease, first the symptom is inflammation. Any disease conditions, inflammation is a major symptoms. Without inflammation, there is no disease developing of in our body. So here also we are looking for antibodies with the biomaterials. Biomaterial means mainly within the natural substance or natural molecules. So when we are looking, so especially Karanjin, I am using Karanjin molecule from the Karanja plant. It's a tree, actually it's a kind of trees. So these projects I received from Ministry of Higher Education, Malaysia. So this is mainly we are targeting of this kind of plant, we are making of some kind of biomaterial formulations. So when we administer to the animals, but we are not completely formulated anything, just we are mixing or soluble with some solubility solvents, then we administer orally. But when we go to the next level of research, maybe it's a solid preparation of some compounds like formulation, is a really fantastic. The world is impact is very high. So when we targeting of this, so we targeting of mainly for adipocytes. So how the adipocytes expanding or expressions. So mainly we targeting of adipocyte cells, we need to block or we need to arrest or we need to reduce the expression of adipocyte cells, then only we reduce the expansion of the adipocytes. So when we targeting of this is our body weight also we can control, also we can reduce our fat, unwanted fats, especially for the white fat. So white fat is very dangerous contains of sugar, contains of too much cholesterol, which is really is a bad cholesterol, definitely affects our cardiovascular system. So most of them, why the problem is coming mainly when affecting of metabolic system, automatically cardiovascular functions also affected. So that is when we are targeting this kind of therapeutic approach, when the metabolic functions normal, definitely we can prevent the cardiovascular diseases, especially for atherosclerosis, the bad cholesterol, LDL. So definitely we can approach, but it's a brown cholesterol is okay, fine, it's very good, it's a good cholesterol, but most of the people, the good cholesterol very less. The bad cholesterol is very high, even though me too, is a LDL is very high, but I'm very controlled, even though I'm never taking of oily food, sometimes I know I'm taking very juicy food, but for me also LDL is very high. So what I did after that, so doctors also afraid, immediately have to take atorvastatin, some statin drugs. Then what I did, change my food pattern or something. Every day morning I take only vegetable or something, some kind of vegetables. Within 3 months, surprisingly, when I went to the blood checkup, the next follow-up, the doctors also surprised, sir, what you did? No, nothing I did, nothing, just simple, simple procedures. Totally is normal. It means the food makes sense. The food is a medicine. When we follow certain small procedures only, maybe some alterations with our normal diet, definitely we can control everything. So this is 2 years I am maintaining of high, high level of LDL, high level of cholesterol. But after 2 years, within 6 months we can control. It means their doctors also surprised. Then I told some hints. So these are the methods, if you follow, maybe we can control ourselves. So kind of why I am telling this, the natural is more values compared to the synthetic materials. So here I followed the natural material only, maybe in the form of vegetable, in the form of maybe fruit, something. But this kind of approach really is very impactful. Maybe we cannot say this one to the international forum, because international forum, they need scientific evidence. By practice, they never accept. 5,000 years ago, is Agastya, they derived is Andrographis paniculata. It's very important, right? 5,000 years ago. Andrographis paniculata is a neem leaf. It's against HIV infections. But this one we never patent or anything, we never bring to the international level. But Germany, they patent. The Germany, they bring the neem oil or the neem have a high potency against HIV virus, human immunovirus. But 5,000 years ago, already our great grandparents already derived, already established, already discovered, but we never bring to the next level of research. But there are many complications, many gaps. We cannot say, there are many factors we never bring to the next level. But we all here, definitely we can bring is certain things, maybe in the next level of research, definitely we can bring. So how to bring the next level? Collaborations, very important. So how the, Akhil sir, we, connected, given the platform, so to connect each of us, maybe we can think, we cannot do many things. At least we can do is start with the minimum, start with the small things, definitely it's achievable. So likewise, we can do many research studies. Without collaborations, you cannot move on modern studies, modern research. Because my lab, maybe I have a sophisticated lab. If any one of you wants to come, okay, I wanted to do some, sometimes if you people, maybe you never get the lab facilities. Okay, most welcome. No need to pay, but yourself you have to take care. You can come to the lab. You can 30 days you can stay. You perform your research studies. You get the research. You get the report. You come back to your own institutions. You can submit your thesis. This kind of collaborations, maybe this kind of opportunities, definitely we can give. But monetary benefit, definitely impossible, because they also have some limitations. But facilities we provide, we ready to provide the facilities. So this kind of collaboration is really important. Why this kind of conference very important? I met is eminent researcher here from Jamia Hamdard. So I met today. Definitely within a month we make some connections, kind of research collaborations. So likewise, this kind of platform really is impactful to do some kind of collaborations, possible way. We cannot do everything, impossible. But at least we start with something. So with this, Introduction to the global view of obesity is a major problems globally, strongly linked with the cardiovascular disease, diabetic and metabolic syndromes. In India is more than 100 crores. Every year they spending on is diabetic induced kidney failure. Only for diabetic induced kidney failure, the government of India, 100 crores every year, 100 crores they are spending several hospitals. So day to day increasing the prevalence of diabetic associated kidney failure. So long time diabetics definitely affect the kidney functions, especially renal functions. So they get complications, they go for dialysis, artificial purification of blood. So there are many complications. So we have to take immediate actions or awareness to the public. So how to prevent? So World Health Organizations, more than 1 billion obese globally. More than 1 billion people are obese. They found more than 1 billion. So with this, in India is a very high. Abdominal obesity is more than 39%. Everybody is very slim, but it's a stomach only. Is abdomen part is a little is a bigger than is other is a body shape. It's a because a lifestyle. Even me too also. The problem is we never control our food habit or something. Maybe many factors. Okay, lifestyle modification, sedentary lifestyle. We never go for any exercise sometimes. Sometimes one day we go, 2 days we are taking rest. So so many issues, maybe work pressure. Environmental factors also very important. So work pressure is a college, I'm also working in university. Sometimes they used to call 7 o'clock. Until maybe 9 o'clock I am working. So that day I go for any physical activities. So India also same maybe, but we need to find sometimes. Otherwise you bring your, the exercise kit to the university. 1 hour break you can go. This kind of activities in Malaysia they are doing. They always bring their exercise kit. Whenever they have free time, they go for gym. Every university they have gym in Malaysia. So this kind of activities, we have to create ourselves, recreate ourselves. Nobody give platform for us to do anything. So we have to create our own. Okay, with this obese rate in India is a 5.21%. Is in India, the throughout India, especially in the South India is very high. South India, so obese is very high, especially Tamil Nadu and also Andhra Pradesh, Bihar, certain area is very high. High prevalence of obese. Maybe lifestyle, it's a food habit, maybe the food habits. So these all, many factors are involved, not only for food habit, genetically there are many linkages. Even the obese also not only for lifestyle. 90% is a lifestyle, but 10% is a genetic linkage also there. So these kind of situations, we need to go for novel drug developments. It's really very helpful for the future generations. With this hypertension prevalence, diabetic prevalence, the key point is most of the Indians metabolically is unhealthy because in our gut system is almost is a 60% damaged. Those who are adults, more than 40, age of 40s, almost 60% the normal gut system affected because of the food. So everything is a chemical. Whatever you take the fruits, whatever you take the vegetables, everything is contaminated by the chemicals. Without chemical, none of the plants also grow. None of the seed also never grow also because the genetically they modify. So many complications. With that only, we have to protect ourselves. So with this all, we cannot avoid certain things, but we minimize certain things, especially for lifestyle, food habits, routine exercise. This is very helpful for us to move on to the next level. Current management of approaches, lifestyle is very important. How the lifestyle is a diet control, exercise, the behavioral therapies. So this kind of is a non-pharmacological approach without medications, non-pharmacological approach which is very important. Pharmacological therapy is appetite suppressant. There are many drugs are available to reduce the appetite. Why you want to reduce the normal appetites? Again, some complications. Indirect effects. Something is a direct effect, something is indirect. Internal body organs also totally the normal functions also changing sometimes. When keep on changing of our normal immunities or normal defense mechanisms, is too much complications in the future. That's what many cancer why developing, because of our normal immune system totally we are changing different ways, by food, by drugs, by other environmental factors. So pollution also is very important. The tuberculosis in India is a number one in the world also. Tuberculosis is mainly for pollutions, a different type of pollutions. We don't have any control, or we don't have any preventive or precautionary regulations. There is no SOP. We never follow any SOP. Antibiotics we can take anywhere in the in the pharmacy, but in Malaysia you cannot buy single antibiotics without prescription. With the prescription only you can buy. But here you can buy any antibiotics without prescription. Amoxicillin, cloxacillin, clarithromycin, azithromycin, roxithromycin, any antibiotics without prescription, without OTC we are buying, over the counter. So these all problems, sometimes the over the counter itself making so many issues. So these all regulatory bodies or regulatory approach we need to tighten. When we tighten, then only we can reduce all side effects. When unnecessary we are using for inappropriate antibiotics, that itself making so much complications. But we do not know. At the time of maybe you can get relief, but long term effects is very heavy. You cannot take any other medication, sometimes treatment failure, cancer progressions. Nowadays whatever we are eating for non-veg, like including for maybe non-veg items, fish or chicken, almost contaminated by the antibiotics. So these antibiotics when transmit to the human also become resistant. So if the cancer patients, definitely the chance is very high triggering of worsen conditions of cancer. So nowadays why complicated for cancer drugs? It's very important. Many people are telling you, our drug formulations, different types of drug formulations, silver nano formulations like a silver, gold, different types of nano formulations. But why still complications? What is the gap? Because our body is constructed by the atoms, like a basic with the atoms, molecules, cells, tissues, organs, systems. So our drugs mainly superficially only acting, maybe system level, maybe organ level, but at molecular level none of the drugs maybe is not acting. That's what is still complicated with the cancer treatments. When the drugs penetrate into the molecules like a DNA or RNA, definitely treatment is very easy. Lot of money we are spending on cancer research, but still we are facing so much of problems for the development of cancer drugs, because the drugs may not penetrate into the molecular level, not reaching. Maybe drug delivery is very poor, reaching to the molecular level, but the cancer developing from the molecules only. The cancer mainly progress from the molecular level, developing from the molecular level like a DNA and RNA level, but drugs never reaching to the RNA or DNA level. So these are the complications. Maybe the future research, especially they are doing for pharmaceuticals research, drug developmental research, is really the biomaterial compounds maybe is enhancing of drug delivery to the molecular level. So then it's a very easier to identify or to develop the novel compounds for the cancer treatment. Still the many drugs, Pfizer, many companies are working on this, but still there are many complications. Solid drugs, nothing. There is no solid drugs for the cancer patient, but depends on the conditions, depends on the stage, depends on the cancers also. Maybe more than 100 cancer types. Okay, limitations for the current treatments is mainly for the same like nausea, vomiting, dependency. Sometimes when they're taking of medications, the day is fine, very good. They feel very comfortable. But once stop the medications, they feel very tired. Psychologically, they have symptoms, or without medications, they are not feel well. So these are the complications. Bariatric surgery, like when the patient with the obesity, they go for surgical procedure, but lot of complications in the future. So these kind of complications, how to avoid? So with the new drug developments, especially for the biomaterials, really is very helpful. Need for innovative solutions. Everybody talking about innovations. Everyone talking about novelty. So, but really we need to go for some novel compounds or novel drug developments for the metabolic dysfunctions, especially for the obesity. The obesity rates is climbing is both developed and developing countries. Before that, we are thinking only for developed countries, USA or maybe Germany or any other Western countries. But it's not the fact. Even poor countries also developing very high. Is a Somalia, maybe is a Congo, is a Nigeria or Ethiopia. So nowadays, because when they modernizing, lifestyle modifications, definitely the metabolic dysfunctions happening, is obese level also increasing. So this is mainly for poor lifestyle. Poor lifestyle. Poor lifestyle means is not by the dress. It's a poor lifestyle mainly by food habit, their lifestyle. So when changing of this, so really there are many complications. So we are looking for sustainable approach. So everybody talking about sustainable approach. Sustainable means is mainly the long-term approach. Is without affecting of ecosystem. Without affecting of ecosystem. Mainly our university we are focusing like Faculty of Medical and Life Sciences, we are focusing SDG 3, health and well-being. SDG 17 is a collaborations. So kind of sustainable development approaches which is really is very important. Then only you can get real advancement techniques. Sometimes within our university we don't have much facilities. We don't have modern techniques, modern machineries. So really is a collaboration is very important. When we collaborate with the good universities or really doing the research intensive universities, really is very helpful for us to learn something, the new techniques, new advanced research studies. So applications mainly for orthopedics, bone grafting also, wound dressing. There are diabetic induced wound also we can use for biomaterials. There are lot of drug formulations we can develop, which is very important. Nowadays they more focus on biomaterials kind of formulations. So new applications regulate appetite and biodegradable polymers can encapsulate the drugs mainly for the slow and sustained release. So mainly we are looking for slow and sustained release drugs only, because then only the duration of action is also increasing. So immediate release, sometimes the duration of action is very short period of time. When we give slow release into the body for the longer period of time, maybe 24 hours, 48 hours, still there are some drugs, fluconazole, antifungal agents. It's a 48 hours, 24 to 48 hours, right? So certain drugs are still we have a sustained release tablet, but mostly we are looking for long-term releasing drugs. It means when the slowly release of the drugs, maybe the therapeutic response also we can extend. So this kind of approach through the biomaterials, not with the chemical compounds. So which is very important. Polymer-based drug delivery, especially for the GLP-1 analogs. There are many, many examples. Just I am given for only one example here. Bioactive composites. So this kind of composites is a really within the present the bioactive molecules, which is without affecting of ecosystem, is a purely is a natural compounds, can influence the appetites, regulate the hormone. Most of the biomaterial does not affect the metabolic functions, only regulate the metabolic functions, or maybe normalize, never decrease, never increase, only normalize the functions. So this is our approach. The hypertensive drugs never decrease your the blood pressure, only is normalize or is a maintaining of normal blood pressure. So likewise, our approach is mainly for regulating of normal metabolisms. So with the biomaterials, we can do many things. This is the initial stage. Maybe I am not going to much on formulations, because my area is a pharmacology. I am doing only with the animal modeling. Whatever I have drugs, now I am doing with a current gene molecule, which is available, is a natural compound. I purchased from India only, from Andhra Pradesh. So there are some compounds when we hear, they never done with the obesity. They done only with the inflammation, some kind of other activities. So I am looking, so why not we go with the metabolic studies? So this kind of approach I am doing. Suppose if you have any ideas, okay, this is the fantastic compound. Really have some linkage with some pathways, AMPK pathways, AKT pathways, maybe NRF2 pathways, ROS mechanisms. These drugs definitely link to some kind of this mechanism, inflammatory pathways, most welcome. Maybe we can collaborate, we can find specific pathways. through the biochemical estimations or through the animal modelings. So I am also looking for certain pathways through the animal modelings. So influence remodeling is very important. So how we targeting of adipocyte cells, especially the brown is very dangerous, brown adipose tissues, which is the bad cholesterol, bad fat. Mainly the biodegradable polymers can release anti-obesity, peptides, natural compounds, mainly for the controlled fashions. So these all very important, but I cannot go into the deep of what I am doing in my laboratories, just for general introductions only. Implantable biomaterial devices, there are many chips nowadays in IIT Bombay, IIT Chennai, they are doing kind of chips, small chips, nano chips. With that also we can deliver the drugs. There are many new technology, new advancement, but within the formulations cannot go to the next level. When we integrate with the technology, then only we can move to the next level. So whatever we prepare any formulations, better we try to integrate with the artificial intelligence. Nowadays there are machine learnings, also some technologies really is enhancing of treatment approach. So this kind of approach is really needed for among in the institutional research, not only for company research, the institutional research also is very important. So with these advances and advantages of biomaterials, reduced systemic side effects, why needed for this research, better patient compliance, to give the better the treatment outcomes to the public. The precision medicine, precision is everybody knows, targeted treatments, specific locations. Okay, this kind of approach is very important. Integration with digital health, which is very important nowadays. Digital health is very important. Even when I was working Monash University, the engineer working with the pharmacy department, the engineer working with the medical faculty, because they only know the technology. They only know the digital technology, how the microchips, how functioning of microchips, how we can go to the infuse, how invasive, the small invasive procedures or open surgery, they only knows. With a small wire, they can insert the chips into the internal organs. We can easily monitor. So there are many technology we do not know. Basically, we all pharmacists, sometimes the technology is we all behind. So when we integrate with the engineers or maybe other professionals, we can get a lot of ideas to move on to the advanced research. So with this digital health, we can easily monitor how the blood sugar, nowadays already available, blood sugar monitoring, maybe the cholesterol level also we can monitor, maybe in the future research. Also personalized therapy, which is called as precision therapy. Nowadays we are going for genetic profiling. So let's say 4 cancer patients, for the breast cancer patients, same treatment, but everybody may not get same response because the genetic difference, individual difference also there. So that we are looking for personalized therapy. So genetic profiling, we go for genetic profiling, how the genetic sequence, how arrangements, maybe is there any genetic linkages with the specific patients or particular patient. So with that they go for the treatments. So genetic treatment approach nowadays is very famous also in modern countries, especially Western countries. So this kind of approach maybe is enhancing of our research studies. Even animal modeling also we can implement. So we can go for a profiling, animal genetic profiling. So with that maybe we can induce cancer, maybe we can induce the obesity. So how the sequence altering with the compared to the normal animals. So this kind of approach maybe is very helpful. We can get help from Sutix people. We can get help from maybe the chemistry. Everybody is important, right? To enriching of research. So with this case studies, so an emerging evidence, preclinical studies, which is very important. Without animal modeling, many people are telling reduce the animals. Okay, only we go with the software. Yes, correct. But when we approach to the clinical trials without animal modeling, they never accept our proposal. So minimum animals, fine. Minimum animals we can take, but animal research is mandatory. Then only we should know the physiology, mechanism, how the physiology, how the drugs are responding to the living substances, particularly why we are taking always for the mice or rats. Why we are taking? Because it's a 90% similar to the human being physiologically. So that then only we can understand the mechanism, physiological mechanisms. Then only we understand the toxicity. Without doing for animal modeling, sometimes it's very difficult to understand. Maybe we minimize the numbers. Before we are taking for maybe each group, maybe 6, now 3 is sufficient for toxicity studies. Okay, so this kind of minimizing can be we can done, but at least we need to perform some studies. Then only we can identify. Maybe sometimes the preliminary level, maybe we can go for zebrafish. There are many research studies, especially for the brain disease, Alzheimer, Parkinson. I think it's Jamia Hamdard, they have very good facilities. So here itself, sometimes we are looking for outside, but Jamia Hamdard is a #1 university in pharmacy. You know, better we collaborate with is a Jamia Hamdard, get the exposure, learn something, grab something. So this kind of interest we must have. No need to hesitate to approach anyone. Sometimes, you know, immediately they never accept. If we approach 10 people, 9 people they denied, but at least 1 people they accept. So when I approach, many people they deny also, but at least 1 person is accepting. So we go with those who are accepting. So at least we move to the next level of research studies, so which is very important. Biomaterials is really very important for fat research, especially for obesity. Future directions, regenerative medicines. Mainly we are looking targeting of adipocytes. How the adipocyte cells maybe expression is reduced, automatically the obese level also reduced. So how to control? With the engineering technology, maybe we can go for some kind of therapeutic approach. Gene editing, which is especially for gene editing, biomaterials, targeting metabolic approach or metabolic research, which is very important to move on to the next level of research. AI definitely is artificial intelligence driven biomaterial designing to get some ideas. Before that, we always looking how the docking studies, fantastic studies. Nowadays, my students always, I start with the computational studies, in vitro studies, animal studies. Because the computational study, without money, without much expenses, we can get lot of data. Okay, how the protein bindings, how the gene expressions, how the receptor affinity, very easy to find. The chemistry people here, they are all working with the docking studies. So at least fundamental something we should know. Maybe we can get some support, but the same thing we cannot apply for the clinical approach, but at least we get some results. So this kind of studies really is very important. The computational modeling followed by in vitro studies and is in vivo studies really is helpful to move on to the clinical applications. Challenges, definitely everywhere challenges are there. Any research without challenge, nothing. But challenges, regulatory affairs, a lot of procedures, we need to submit evidences. Without evidences, they may not support you. Sometimes they never listen to you, your ideas. If you go and approach to anyone, they're never interested sometimes. But keep on, we have to prove ourselves. It means we have to show the evidences, our lab research evidences. Then only sometimes they may agree. Without this, they may not agree also. Sustainability is very important. Sustainability means it's a long-term approach. It's not one, it's a, whatever we develop any drugs until their lifetime also should not produce any toxicity or any side effects without harmful for the internal organs. So this is very important, so that we always looking for the SDG 3s, good health and well-being. So also collaborations, it's a mainly for SDG 17. So this kind of approaches really is very important for us to move on to the next level of research. So with this, biomaterials is a represent a transformation of next level of research, offer a safe, precise therapy and also long-lasting treatment approach. Also potential to replace conventional management or conventional treatments. We can replace, because with the conventional, lot of drugs already we withdraw from the market. Example for nimesulide. The nimesulide no more market in the USA, already withdraw from the market. There are many drugs already withdraw after 20 years, after 15 years using from the population, from the public. Why? Because lot of side effects. Sometimes the drug itself to damage of our heart muscles. Lot of complications. So that now we are looking biomaterials without affecting of our internal organs or internal tissues or any other parts of our body. So we are looking for novel approach. With this, I thankful to Ministry of Higher Education providing me the funding opportunities to carry out this research, still ongoing. So our pillars of research success is mainly for research collaborations. Without collaborations, is one hand is really is very difficult to make sound. So when we keep many hands, definitely is a more sound. Also we can better research outcomes also. Individuals who contribute to the research process is very slow, but when we together we go with research studies, definitely outcome are also good, effective outcomes. But we cannot go with everyone, it's impossible. But at least wherever possible we can align. Also the funding opportunity is very important. Without funding we cannot do any research. Research means we need fund. Without money cannot do any research. So we try to find international funds. We try to find DST fund, DBT funds, ICMR funds. That's what yesterday many people came from ICMR and CSIR. So we approached them. So is there any opportunities to give some funds? Of course, Jamia Hamdard, Galgotias University, they all obtain many grants, maybe from the ministry or different, government of India as different bodies. But that is not sufficient. Maybe international grants also we can approach to get more funds to move on advanced research. And institutional collaborations also is very important. The infrastructure also very important. Sometimes admission is very good, but the infrastructure is very, very less. There is no small instrument also. There is no centrifuge machine. There is no HPLC. There is no mass spectroscopy. There is no basic instruments. So we need to build up the infrastructure. Then only the student get interest. Also the lecturers also how to give the protected time. Maybe the dean sir, please give the protected time for the staff to perform. This kind of approach really is very important to move on. Always is a Malaysia also. The protected time really is very important for especially for the researchers. When we get the protected time, definitely we can move on to the next level of research. With this, thank you everyone for listening my presentations. Thank you. Finished?
Thank you, sir. Thank you so much, sir, for this insightful presentation. Thank you so much, sir. And I would like to add that you can join us here physically from the Malaysia, and we are really thankful to you. So thank you so much, sir. Now I would like to call sir on the stage. We want to felicitate you, sir. And for the felicitation, I would like to call Professor Dr. Zeenat ma'am from Jamia Hamdard to felicitate the sir. Everyone have a big round of applause. Thank you so much, ma'am. Thank you, sir. Now it's time for the high tea. Everyone, please go for the high tea. And within 15 minutes, please reassemble here for the next session. Oral presentations are already going on. Please, candidates, go for the presentation. If anyone is remaining for the presentation, go in the boardroom. Presentation is going on in the boardroom. Before going for the scientific session, I would like to call Dr. Fawzia Zakir, Assistant Professor of B.Pharm Ayurveda at Delhi Pharmaceutical Sciences and Research University. Ma'am has worked as woman scientist under DST, over a decade of teaching experience, and with more than 1000 citations and more than 50 publications in high impact factor international journal for their presentation. So ma'am, please come on the stage. Everyone, please give a big round of applause to the ma'am.
Thank you so much. Presentation mode. Good afternoon, everybody. So I'll be discussing about my work, which is reformulation of an Ayurvedic agent and how reformulation into a nano delivery system helps in enhancing the therapeutic outcomes of an Ayurvedic medicinal agent. So the talk I'll be discussing here is for the treatment of epilepsy. Now I won't go into details. We all know what epilepsy is, and we all know that it is a major neurological disorder which is currently affecting more than 80 million population globally. So there are various therapeutic agents which are available in the market. Regrettably, these therapeutic agents suffer from a number of side effects, and in some cases, patient can even experience CNS impairment and even death in extreme cases. So talking about Ayurveda is a traditional medicine system which dates back to more than 1,000 of years back, and we all know that it is reported to be safe, it is efficacious with minimum side effects. So as per Charak Samhita, it has been reported that Shatpushp oil, the Shatpushp thylam is an Ayurvedic medicinal agent which is obtained from the plant Anethum graveolens. Now, this medicinal agent has already been reported that it can be used intranasally for the treatment of epilepsy. Of course, it is very efficacious, but there are, again, various challenges. One of them is this oil is extremely irritating in nature. When it is administered intranasally to the patient experiences extreme irritancy, and because of which it makes it extremely inconvenient and discomfortable for the patient. And because of this, patient is usually discouraged, and he discontinues the treatment in between. Another problem, it is an oil-based medication, so there are chances of mucociliary clearance and poor residence time in the nasal cavity. So our research envisaged is to develop a thermosensitive nanogel for intranasal delivery of Shatpushp thylam, so that we can enhance the residence time, enhance the bioavailability, and at the end, we can enhance the therapeutic outcome. So our vision was to develop a nano-emulsion delivery system. Because it is an oil-based drug, we successfully incorporated into a nano-emulsion, where using different approaches. By using aqueous titration method, we used a range of surfactants and co-surfactants, and we successfully developed nano formulation with an average size of 119 nanometer. And it was thermodynamically stable. Of course, we have done all the physicochemical characteristics. Our next objective was to develop a thermosensitive nano-emul gel. Now, the purpose of developing a thermosensitive nano delivery system was to convert it into a gel, so that it is actually an oil-based formulation. So when we use thermosensitive polymers, it is converted from salt to gel form instantaneously within seconds, as soon as the formulation is exposed to a temperature of nasal cavity, which is 29 to 30 degrees Celsius. So as you all know, oil has a chance of poor residence time, but when it is converted into a gel form, because of its mucoadhesion property, because of the use of polymers such as poloxamer and carbapol, we can convert it into a gel form which has prolonged residence, prolonged mucoadhesion in the nasal cavity, so that it gets sufficient time for permeation from the nase to the blood-brain barrier. So for that, we used a range of polymers. We have optimized different concentration of polymers, and we have optimized the gelling temperature, the gel strength, as well as the gelation time. Now here, gelation time is very important. It should be instantaneous, so that it does not get time to drip from the nasal cavity. Next, we performed nasal permeation studies we have compared the permeation of Shatpushp thylam with plain Shatpushtalam, and we found that the formulation enhanced the permeation by approximately 8-fold. We then went on to perform pharmacodynamic studies where anti-epileptic property was tested, and we have found that the formulation successfully reduced the seizure duration by approximately 76%. Now, the reason for enhanced permeation and better anti-epileptic property was obviously because of the nano size of the formulation, increased residence time of the formulation, so the drug was able to permeate from the nasal region to the blood-brain barrier. And obviously, because of the use of different surfactants which act as a penetration enhancers, which have provided better delivery, improved permeation. Now, of course, as I said, Shatpushtalam is extremely irritating in nature, and on top of that, we have used different surfactants, which again are irritating in nature. So we had to perform nasociliar toxicity studies. For this study, we exposed the nasal tissue with our formulation, and we tested it for histological, we compared it for histological changes, and we also observed it under scanning electron microscope. And we found that when we expose the nasal tissue to a positive control, which is isopropyl alcohol, which is an irritating nature, it disrupted the nasal epithelium. But when our formulation was treated to nasal tissue, the cilia remained intact, which suggested that it is a biocompatible formulation. And this was, again, it was proved by observing the images under scanning electron microscope. As I said, carbapol is a mucoadhesive agent, so we tested the mucoadhesive property using texture profile analysis, and we observed that it possessed sufficient mucoadhesion with a mucoadhesive force of 0.2 newtons. Now, this is important because, again, if we are using very high concentration of carbapol, if it is extremely mucoadhesive, again, it can be irritating to the mucosal membrane. So it should be within a limit. It should be usually less than 0.2 newton, which suggests that it is safe for use. So overall, during the study, we have found that we were successfully able to incorporate Shatpushtalam into a nano emulsion delivery system, which has successfully enhanced the permeation through the nasal epithelium by approximately 8-folds. It was a biocompatible formulation, and we have tested it for various physicochemical properties such as thermodynamic studies, physicochemical studies, physical stability studies, and in the end, we also performed pharmacodynamic studies to compare the anti-epileptic property of the drug encapsulated in nanogel, and we have found that when it is encapsulated into a nano delivery system, it successfully enhanced the therapeutic outcome. So with this, I think in brief, we can conclude that whenever we reformulate an Ayurvedic agent into a nano emulsion delivery system, there we can successfully enhance the therapeutic outcome. And I think this opens the doors that how different Ayurvedic, there are obviously the various challenges with the use of different Ayurvedic medicinal agents. So we can work upon that, and we can improve the therapeutic outcomes and, of course, the bioavailability and the patient much, and I would like to conclude. No, this oil is actually a one component. Shatpushtalam is actually, there is a reported procedure to prepare this formulation. That is, we have to take oil, the leaves of the plant, and we have to boil it in water in a ratio of 1:4, and we have to leave it for boiling overnight. Till in the end, we get an oily residue. Actually, we did not perform this formulation. I got this formulation from All India Institute of Ayurveda. This is a collaborative work which I performed in collaboration with them. They approached me, they gave me a product, and they said, this oil has this problem. Can you overcome the problem of irritancy and poor residence time of this formulation? So I gave them an idea that we can reformulate it into a nano emulsion delivery system, which has a very good loading. Nano emulsion, the major drawback of nano emulsion is its scalability is very good. And another advantage, it has 100% loading efficiency. That whatever drug we are using, there are no chances of wastage. There is complete encapsulation of the drug. And obviously the dose of Shatpushpthylam, it is already reported in the literature in Charak Samhita, it is reported also in Ayurvedic Pharmacopoeia of India also, it is reported. So we have used that dose for the preparation of the formulation. Yeah.
Amazing presentation. Mainly when animal modeling, when you go for animal modeling, how you administer the drug, what kind of needle you are using?
Actually, there is a separate device which is available for instilling the drug into the nasal cavity. But what we have used is we have used a micropipette. Because in micropipette we can optimize the dose, that how many microliters of drug is going into the nasal cavity. So for that purpose we used a micropipette with a capacity of 200 microliter, and we instilled 200 microliter of the formulation into the nasal cavity of the rats.
So while you administering the drug, is with anesthesia?
With anesthesia, obviously.
With anesthesia.
Yeah.
Very interesting. But the oil formulation, is it appropriate for for nasal administration?
Yeah, it is. In fact, in Ayurveda, this oil is already being used for epileptic patients. But the main problem is, as I said, it is very irritating when it is instilled into the nasal cavity, because the Dr. Pramod Yadav from All India Institute of Ayurveda, he approached me. He said that I am giving this oil formulation to my patients in the OPD, and anti-epileptic patients are approaching me. But when I give them this drug, they said that this is very effective. The problem is they don't use it. They said that it is very irritating. We can't use it. They will use it maybe once or twice, and then they will stop using it. So,
How are we going to modify?
sorry?
Because when we modify, okay, this is effective for patients, right? So how to modify? So what measures maybe, is there any possibilities to make without irritations?
No, that is why I said I have reformulated into a nano AIMIL gel. We have incorporated it, and we are using it in combination with polymers. These polymers are safe, efficacious, because the Shatpushp oil, that is xylem, is encapsulated into a nano emulsion. So when it is encapsulated, obviously the drug is not in direct contact with the mucosal membrane, so it is obviously no irritation. Yeah, I have done nasociliary toxicity studies and scanning electron. You can see here in the image, you can see the cilia, brush border cilia appear. That should remain intact. Yeah, tissue. Uh, yeah, I don't have it, but okay, sure, sir. Huh. No, that is what we want. It is, no, actually, it is bypassing the brain because of the nano size. No, it is reported that if you want the formulation to cross the blood-brain barrier, first of all, it should be a lipid-based formulation. And another thing is, the size should be less than 200 nanometer. First point. And the second point, how we ensured, how do we know that the drug has crossed? Because we want the drug to cross the blood-brain barrier and reach the brain so that the drug can act at the receptors there. So that we have proved. We have done animal studies and we have compared. You can see when we are using plain Shatpushpthylam in pharmacodynamic studies, you can see the efficacy is much less, but when you incorporate it into a nanoemulsion formulation, the anti-epileptic property has significantly improved. That is because of incorporation into a delivery system. Because of the nano size, it is able to permeate the blood-brain barriers, and obviously there are no chances of loss or leakage because, as I said, it has 100% entrapment efficiency. Whatever drug we are using, all of the drug is able to encapsulate in an nanoemulsion delivery system. And, because it has very good residence time in the nasal cavity because of the presence of mucoadhesive polymers. And another thing, as I said, that we have used a polymer poloxamer. Poloxamer is a thermosensitive agent. So when we are formulating it in the lab, it is a sol form. When we keep it at room temperature, it is a sol form. But as soon as you administer it into the nasal cavity, within 15 seconds, it will convert from sol to gel form. That is the beauty of that polymer. So once it is converted into a gel form, on top of that, it has a mucoadhesive agent. So it remains in contact with the nasal mucosal epithelium so that it gets sufficient time for the drug to permeate through the nasal layer and reach the blood-brain barrier. No, as I said, we have the dose which we have used. We have used the reported dose, which is reported in 200 microliters. 200 microliters per kg. Okay, thank you so much.
Thank you so much, ma'am, for this wonderful presentation. It is great opportunity to have you with us today. And I remember, ma'am, when we have conducted our first conference, CFPPSC 2023, you are the great support in that conference too. So thank you so much, ma'am. I would like to call Professor Dr. Zeenat Iqbal, ma'am, and Professor Dr. Shweta Sharma, ma'am. Please, ma'am, come towards the stage. Thank you so much, ma'am. Of Pharmaceutical Sciences, currently serving as professor in the Department of Pharmaceutical Chemistry, Faculty of Pharmacy, AIMST University, Malaysia. Professor Fuloria has an extensive and illustrious career spanning 22 years across academic research and industry. Sir holds multiple prestigious qualifications including B.Pharm, M.Pharm, PhD in Pharmacy, an MBA and a PDCTM, demonstrating his interdisciplinary expertise. His impressive research contribution include 3 MOOCs, 3 books, 11 book chapters, 15 patents, and 187 research papers, reflecting his deep commitment to innovation and scientific advancement. Sir has successfully secured 9 national and international research grants and has supervised and has supervised multiple PhD and postgraduate students shaping the future of pharmaceutical research. With participation in 126 conferences, trainings, webinars, and seminars, and being honored with 6 prestigious awards, his contribution to the field are truly commendable. Sirs, today's topic of talk is orthomolecular medicine and chromotherapy, the research avenues. Please, everyone, let welcome the sir on the stage. Have a big round of applause to the sir.
You had a screen poster earlier. Can you fix that poster again? Yeah, yeah. See that. Sorry for a little delay. Firstly, I would like to congratulate the P.R.S. Education Trust, Jamia Hamdard, and of course Sunway University to organize such a wonderful platform wherein we all can exchange our ideas, views, and any novel innovations, whatever going on so far. So we are here to discuss. And nextly, I would like to thank all the participants, especially the students, with full patience. They are sitting so far without dropping the eyes, without sleeping. I was continuously observing you all, I was just observing who is sleeping, who is nothing. No, full professional. Very nice, really. Thank you so much. So, before I, oh, here it's a different screen, and there it's a different screen. I want to show something interesting actually. I want to show this screen firstly. Thank you so much. But there it's coming that one. Before I, that screen is not coming. Not there. I want to show something interesting actually. Never mind. I'll do one thing. I'll try my own way. No issue. Print screen. Okay. I'll go to the PPT straight. No issue. This is my slide. Oh, why so? I'll try my own way. Okay. Okay. No issue. I'll go to this. Control M. Hopefully it will come. Okay. Never mind. Something else is coming. Actually, I want to show the banner screen, but here we cannot see. So I thought I'll print screen here, but still it's not coming. this how to see? Okay. For a time being, he'll be here only. Just a moment. No issue. You don't go. Don't go. It's okay. Actually, why I was interested in showing this banner? You can see the pictures. The first, second, third, fourth, fifth, sixth. That person is there. His name is Professor Neeraj Floria. This is me. Although now here we are two different persons, so you may have a complaint. The person is different over here. Some game has happened. No, I'm the same person. So thank you. Now I'll come to my presentation. Thank you so much. Sorry for this short clip only. I was about to speak something. Now I want to come. I want to come on a point why this difference is there. Yes. Thank you. Thank you so much. So reading will be difficult. Can you put not full screen? Oh, then I have to wear my specs. Okay. So coming on to the first screen, what we had just now, you know, that's a miracle. It happened because of some topics going on since morning. The first lecture was natural molecules or natural medicine. Second topic was biomaterials, that again natural. The third topic was also again Ayurveda. Because of some topics going on since morning, the first lecture was natural molecules or natural medicine. Second topic was biomaterials, that again natural. The third topic was also again Ayurveda. You know, the objective behind. So here, it's a new idea which I am throwing in front of you. It's orthomolecular medicine. In many platforms I had discussed earlier also. But here I am talking two ideas together today, wherein all the ecology, all the chemistry, Sutics, all can find a new research avenue with some new innovative idea, which in India, I don't think so far, maybe one or two people are working. Otherwise, in India, it's nobody is doing so far. So it's an entirely novel field. So please listen carefully. I'll try to finish in short. Slides are 138, I think. But don't worry, I'll not speak full. I'll give a gist of it. So are you all with me, students? Okay, thank you so much. So the first is, I'll be talking about orthomolecular medicine and brain health. Sorry, have a habit of looking there, so I'll see here now, no issue. So what is orthomolecular medicine? Orthomolecular medicine means, in Greek, ortho means natural. Anything which is natural to our body. When we say orthomolecules, molecules that are looking there, so I'll see here now. For example, we have insulin, we have all the vital amines, we have all the fatty acids, good, bad, whatever. So that is all about I'm going to talk. In short, I'm saying, so that now will come on a little brain health. We say our brain is healthy when? When it is, we say when it is functioning well-being, when we are able, we have the cognitive abilities, emotional balance, and blah, blah, many things. So ultimately, in a good health brain, we say a well-being brain is a healthy brain. Now in today's scenario, different factors affects our health. The main is, of course, our poor lifestyle. Example, when we talk about Nokia time or before Nokia time, we could remember all the mobile numbers. Now, do you remember your friends or anybody's number? No, cannot. Even your number also you have to search sometime. So, it's a poor lifestyle what we have. So, it includes our eating habits. That's a pizza, burger, you know. I'm very good, very sincere, very honest orthomolecular professional, I can say right now. I didn't had all these things. I came here nicely. They offered tea, coffee, milk. No, I'll not take. I don't say don't take. But everything in a limit, okay? So then lack of sleep. Everybody of us normally sleeping by 12, by sleeping by 1. You sleep by 1 and check your cholesterol level tomorrow morning. It will be high. One night shot, it will be shooting. Okay? Then our sedentary lifestyle, lying down, no, you know, earlier we used to eat down on a ground. Now no more. on a chair nicely. That's why all gastric troubles. So I'm very cautious. That's why I was not taking anything, only water. Hydrotherapy, that is one of the components. Then, of course, chronic stress from a longer time. Many of the people yesterday, I was discussing to one of the professor, anybody, today they say two things. When they want to be in a job, for the student's sake I'll tell, or anybody who's in a private job, एक बार government job मिल जाए, उसके बाद, correct. Why? Chronic stress. Stress is there. So another is, like in our, we have a KPI now, everybody got a targets. We all have a targets. So that's, stress is chasing us. So that's why everybody target today, let it be Jamia Hamdard, let it be a Dipsar, anybody. They want to retire in the age of 55. Almost from 50 onwards it starts. We all think, just एक बार बच्चे का graduation हो जाए, masters हो जाए, job पे चिपक जाए, after that I want to be retired, isn't it? My brother-in-law, my sister, they all were in a job. One was in a private job, sister was in a government job. They tell, I want to be retired. Just my son, my daughter, they get occupied. You know, examples. Then some medical conditions. I have a ligament surgery. I had my hip was cutted once. I mean, the full, there was a tumor. They removed. Then I had epilepsy. I had diabetes. I had cholesterol, blood pressure, everything, all sort of diseases. But now today I am okay. Much better because of something like natural medicine. Okay. Then another factor for this brain health, not only the brain, I'll say the whole body. Environmental toxins. Delhi, the best place to have all the environmental toxins. What I heard so far. Okay. Then your trauma, whatever, so many things. Then what are the approaches to, maintain a healthy lifestyle? It's not only brain, full healthy life. One is the lifestyle approaches. Like Dr. Vetri, today morning, he went nicely for a walk to the India Gate. He told Delhi is very good. I said, you have seen the one face of Delhi, India Gate and all. You have not seen the entire Delhi. You cannot go there for the walk in the evening and the morning. Then some cognitive approaches, some environmental and holistic, and some medical and preventive medicine, where I'm coming now, is one orthomolecular medicine, with which we can maintain a healthy life, a healthy body. That was National Center of Complementary and Alternative Medicine, NCCAM it was. Now it is no more under NIH, it is as a integrative health. Now we cannot use the word alternative medicine. So we have the different domains. Domains are still same. In that we have alternative medical system, mind-body, biological-based therapies, manipulative and body-based method, and energy therapy. So this orthomolecular medicine is categorized as a biological-based therapy. Now, why we are going for these domains? The question comes, why we need to go through this? Tomorrow, when you want to apply for some research at a graduation level, at a master level, at a PhD level, let me be very clear. At a graduation level, you want to apply a grant, you can get it. Probably not in India, not in some Asian countries, but Western countries, being a graduate also, you can get a grant. One example, in Malaysia, one time in the 2014 time, we all were applying for a grants and all. One 12th class student was there. His project was very worth. From natural medicine until clinical trial, he proposed the proposal, and all the projects were blocked. They sanctioned only to that boy. So that's why what I am saying is tomorrow, because B.Pharm student also here, or maybe anybody might think, what is the use for us? See, such a big research going on. What we will do with this? This is the area we are in, because it's a plain Ayush ministry is there. You can apply for any number of grants. You will get it. Because nobody is applying for this. That's a separate stream. If you apply, let it be a private government, because you will be the first one to apply. Here, I think the capacity of 50, 60, only 50, 60 applicants will be there. So it doesn't matter. Still, you have a chance to get this grant. If not here, apply Germany. Wherever you have, you can apply anywhere this orthomolecular medicine. So, sorry, one more field is energy therapies. In that, I'll be coming on chromotherapy later. That's another field. So I thought when I came here, I must discuss so that we can have some new innovative idea wherein we can apply all this science. So what is the main aim of this branch? Let it be a chromotherapy, let it be a Ayurveda, let it be any field. We have ultimately to restore the optimum environmental of body. How? By correcting the imbalances or deficiencies in our body. How? By using some substances which are natural to our body. Like example I told, give insulin, restored. Vitamin C, scurvy you have, okay, give vitamin C, replenishment, you will be treating your disease. Like example, one big example, in Corona time, a lot of people were taking vitamin C. Why? Because one of the orthomolecularists, he did a research, because already it was known vitamin C on a high dose gives an antiviral property. So they did a trial and they were having the good results. So this is how, in orthomolecular medicine, in gist I'll tell you, we give the normal ingredients in higher dose. Then the question comes, how we can give in a higher dose? It can be toxic to us, so many problems. Yes, that is our part of study, to analyze for a pharmacologist, for a formulation development, so that in a good formulation, such a way that in a low dose it will give elicit a high response. There comes the responsibility of a pharmacy researcher. We can apply our knowledge in this field. Some examples are like vitamin, amino acid, hormones and all. A little brief about history. You know, you will be very wonder. It's a 60 years old branch, that's all. And the man who started this, initiated the concept first time, he is a double Nobel laureate for this, Linus Pauling. So it's not like that. Today you do some good wonder research. Chromotherapy, another person was there in 1903, he did some study on a lupus, some disease was there, I forgot the name. So there he got a Nobel Prize. So you drag anything, okay. So he was the man in 1968 who introduced the concept of orthomolecular psychiatry first time. Later he got the Nobel Award. So what is this concept of orthomolecular medicine basically? No one size is fit to all. So concept of bio-individuality. Like paracetamol, we give to anybody 500 mg or 800 mg, 600 mg, 300 mg, whatever, 170 mg also. It's common to all. But in orthomolecular medicine, the concept is we do titration because it doesn't follow RDA. RDA problem is, that is the recommended allowances is for healthy professional, not for a disease state. So orthomolecular medicine practitioners never follow RDA. They titrate their dose. They do the trial because for every patient, like in a homeopathy we do, the dose is different. Same way here also. But here, the thing is, things which are natural to your body. Like example, just now madam told some nasal oil, what you have used for the epilepsy condition. Formulation development, you can go for this also. See the disease condition, develop a formulation for this. Like example extension for you, like Dr. Vetri was asking, the idea can be, we can go for nasal spray. If it is a volatile component is there, isolate that, go for a nasal spray, your product is going to be much beneficial. So that's a small ideas. So this is some image I have shown. So OM concept is, every disease can be controlled by increasing or decreasing the nutrients of your body. The natural things of your body, you can control any disease. You are having blood pressure high, control the salt. You are having low, take the salt. Your sugar low, take the sugar. So likewise, whatever is in your body, you have to maintain, that's it. But in this specifically, we never study anything in a higher dose. People say, if we give a vitamin in a higher dose, there can be a, the toxic or, but there is so far, to be very honest, no death is reported with alone any vitamins and all these ingredients which are natural to your body. With medicine, it has been reported, but with this, never. So, for example, some epileptic seizures are there. Depending on vitamin B6, can be managed by high dose of vitamin B6. You can try. So far, not done. But you have to find out whether these seizures are because of vitamin B6 deficiency. So you can go for study. Phenylketonuria can be managed by reducing the high phenylalanine-rich diet. Insulin we are taking always, that is we titrate. According to the patient requirement, we do it. That is we are doing. So this OM treatment, as I told, it varies from case to case. For me, the requirement will be different. For you, the requirement will be different. So vitamin C requirement for me will be different. For you will be different. You need more, I need less, because I am already taking, so no issue. That's why now the concept, actually now what we take a processed food normally. You will take a pizza, cheese, milk you take, processing you do, make a cheese, and after that reheat again and all that processed. So whatever the nutrition is there, on a high temperature, all the amino acid, everything will be gone. That's why in nowadays, in a healthy lifestyle, they say take a steamed vegetables. Not a higher temperature also. Otherwise, if you take on a higher temperature, the ingredients will be gone. In the primitive time, the people don't have the brain disorders or the Alzheimer, the memory loss was not there at that time. If you remember your parents, your grandparents, all that, their life was also, aging was also like after 100 years also. My grandfather, 99. Grandmother, 99. Everybody here just like 99 only. But now, father, 70, 80. That's a common. Us, I already have 4 or 5 surgeries. Example, what I mean to say. So earlier time, the food was not processed one. And you know what is the biggest culprit today? Many were talking about the cancer and all. The biggest culprit for the cancer today is refrigerator. You know why? Because we can easily preserve the thing. After that, we can go for reheating. Example, veg fried rice. Everybody likes. अब भैया जीरा राइस जरूर बनाना. थोड़ा उसमें घी अच्छा रखना. Put some pure ghee, Dr. Vetri. So here they ask put some pure ghee more. So likewise then we eat. But the problem is, one time, if your rice boils, there is a bacillus cereus bacteria is there that grows. You put in a refrigerator, it will multiply later. When you keep it for long on a normal temperature, it will multiply. And then when you heat, there the problem starts. Sometimes many people they ask, person was very good, cholesterol normal, everything normal, how come it happened? He was going to the gym normally, daily, all time, daytime. Problem was food. Reheating food is the issue. So my advice with the orthomolecular medicine, the concept is please look after your lifestyle also. It's not only about the molecules. You have to maintain externally also. Control on your diet. Like example, when I came Delhi, honestly, I had pain started. Water, I was taking mineral water only. I was not touching anything else. Okay. Still also, I was not taking tea, coffee, milk, nothing. I don't take tea, coffee, milk, nothing. I don't say follow me. Follow, but in limit. Some people, they say with a proud, I take every 1 hour I want 1 tea. Not good for health. Sugar. CNS, capacity getting down. Stimulant, you are taking one side. Problem, coffee. So, earlier time processed food was not there. Now we all are taking normally processed food only. The grain, the fiber size, now we need naan, butter naan leke aana. Attack over there in any marriage party function wherever. Roti, who will take? Nobody will take. But UP still, I see many people, they like missy roti. Correct. So, some evidences, Dr. Abraham Hoffer, he was an orthomolecularist. He did some 10,000 more than patients for schizophrenia he handled. And almost with the nutrients and vitamin itself, he recovered. He never gave a medicines, what we give the modern medicine now. He recovered. Likewise, there are some studies, I need not tell about this, I just throw an idea. Google is there, now the ChatGPT is also there. So you can find out, give me the list of studies wherein these many things have been done, you please follow it. I'll not go because I know, I don't want to torture you by reading, reciting one line by line, students especially. Okay? So myth of taking too much vitamin, I told many studies has been done. No deaths were reported with your vitamins or nutrients especially. Depression, the biggest problem today. especially in the Western countries. Here also it is coming now. The format is different, not the seasonal. The another work depression, whatever, your loved one, anybody left you, all those things are the depression causes. So it can be a stress, death of loved one, disability, lack of social support. Biochemical factors, physical factors can be there. Some medication side effects also, it causes depression. Especially the cancer patients, it causes a lot of depression. Genetic factors, other so many factors are there. Especially the biggest factor is because sugar has a property to leach your nutrients outside in the urine, your all protein amino acids will come. If amino acid, everything is going on, your tryptophan, all that will go off. It will affect your brain, depression. I think I need not go in much detail. I'll just go one short study what people have done about, like vitamin A. A meta-analysis was done for the study of vitamin A, where in some 25 studies they have taken, and they have observed like higher dietary vitamin A intake was associated with a reduced risk of depression. When they give the vitamin A, the depression was being reduced. So likewise, if you will Google out, you will find hundreds of studies. Likewise, for vitamin B group as a whole, in this, the psychiatric patients who are admitted to the hospital abnormally have low thiamine level, it's observed. You know, orthomolecular medicine is the only branch in which when a patient comes to a doctor, they check their vitamin level. First of all, they will check what is the nutritional level in a body. Only branch. Other field, pulse, all that, whatever, but nutrition level, they will never check. This is the only the field is orthomolecular medicine. So likewise, thiamine B1, B2 also, some studies are reported for improved energy production. Likewise, niacin B3. I need not go in much detail. So just I want to throw an idea how we can apply all these things into our research. Take any molecule, do its formulation study, do its pharmacological, all the levels, toxicity, whatever, do all the study, apply for a grant, take a grant. But how to get a grant, that's very important. If you will apply directly, nobody will give you, to be very honest. You need to have an orthomolecularist in your team. A person who got a certified license as an orthomolecularist, you will get the grant. Because to diagnose, to study the patient case, everything, who will see? That is the person who is a specialized in that. Then you will get a grant. Otherwise, difficult. So for the students also, you can come with a proposal, make a proposal, go to the lecturer, and then ask them, please settle the team. We want to do it. Because in every research, I think you know or not, we need a student. Without student, none of the government will give the grant. We are bound to give you the master degree, say PhD degree, whatever. In fact, if after B.Pharm or let's say BSc, whatever, you want to go outside India also, you can apply directly for PhD. Even in ICMR. ICMR nowadays, it offers direct PhD after graduation. So make a good proposal, think about, study, Google out, you have ChatGPT, everything, whatever, but don't use the reference of ChatGPT, it never works. I'm telling you today. It's fraud. I'm sorry. It makes the reference with its own. You cannot use that. So Google out, just see, find out, and make a good proposal. Maybe sometime, whenever we'll have a chance, next time, we can have a session how to write the research grant. Luckily, with God's grace, we got some research grant, so maybe we can discuss at that platform. So likewise, this is all about, I need not go in. Likewise, the essential fatty acids like white and brown, what you told. So similarly for that, omega-3 is the main one, what we say. For that, many studies, you can design with your own. Chlorophyll, you see the structure of both the molecules, hemoglobin, chlorophyll, it's same. Only one, that mineral is different, rest all same. So you can do any study related to, whatever is related to your blood, hemoglobin, do with the chlorophyll, okay. So minerals, likewise, many. So I'm going in a, I'm not doing a race here, don't worry. Because I want to give you a gist, I want to give you an idea. What research we should do, because the problem today is, every paper you go to publish, every grant you go to, submission, they will ask one question, what is the novelty? Everybody will ask. So here you can tell, nobody has done in the world this one. So you can go for publication. Or you can go for your study, whatever, the proposal making. You will get the grant. 100% you will get it. But only thing, in India I'll tell, to be very honest with the experience I'm telling, copyright. Copyright your proposal, after that you submit and put a certificate along with your grant. Nobody will copy. We do it in Malaysia. Dr. Vetri, Dr. Manikam Ravichandran and our team, he always say, without copyright will not submit. Because it happened with me in Singapore, somebody, I submitted, not click, but they published the paper. So the word is like that. It's not only India, it happens everywhere. So the key principles of OM practice, this orthomolecular practice is, first is the medical diagnosis and treatment. There should be a sound knowledge about safety and efficacy of the things. Then it is a principle is, which is natural to our body, which is a part of our body, cannot be toxic to our body. Yes, higher and lower dose, we have to decide. Then reliability is, they don't rely on medical reports simply, the practitioner. They rely on your patient trial dose. What is the patient titration dose is, based on that only they rely. So of course, it is based on individuality, the next key principle what we discussed. Dietary allowance, they don't rely on RDA. Because for a disease state, who got a scurvy, for him RDA will be different. RDA never recommends. So they titrate. And of course, environmental pollution also they check. Only branch in which they check the pollution level in your body on every tissue. Otherwise, nowhere in any of the branch they never check. If you go to a doctor, who will check? Nobody. But with the experience only, they can make out what is the complexion, what is all that. Based on their experience, they can tell his metallic construction is how of the body. So this is the same only. So indications, where we can do our research. In psychiatry, that is in schizophrenia, you can work on niacinamide. Already people are doing. You can also do, design some new formulation. Try the study. Depression, people are already working on tryptophan. Dementia, vitamin B12. Congestive heart failure, coenzyme Q10, taurine, magnesium, osteoarthritis, niacinamide, glucosamine, gingivitis, your gums inflammation, folic acid. Likewise, different, there are, I'll tell you, you just go to the net, find out the indication for applications of orthomolecular medicine. You will find thousands of applications. So that means you can make thousands of projects. We always, always are scared. Somebody will take the idea. We should not disclose the things before, otherwise it will go. But here, it's an open field to play, I'm telling you. Try orthomolecular medicine research. So this is one. Now coming to the next. In short, I'll go for this also. Chromotherapy. This is another innovative. You see, one example I'll tell about the chromotherapy is color therapy or a light therapy. Example, newborn baby, got a jaundice. What they do normally? In special nursery ICU, they will put the UV light, they'll put the different color lights that control jaundice. So evidences are there already. Now I'll tell you how to do the research. Take your pharma molecule for jaundice treatment. Take your another ayurvedic anything for jaundice treatment. Do a third treatment with a light. Now add this light to all the three components. Normally how we do the pharmacology experiment with a color, different light. Here you can do on the humans. It is, you need not have a permission also, because it's already people are taking, already commercially available, already they are doing it. Just do connect with the light experiment. It's a new research. Only difference is, here again you need to have one more member who is a specialized in this chromotherapist. So that's just I'm throwing an idea to you, and not necessarily humans, you can do on animals also. All the things, same way. Actually, I'm sorry, because Dr. Akhil told me, sir, please don't take much time. I said, don't worry, I'll try to give the gist what is important. I'll not discuss much. So here it is coming on our fifth domain, energy therapies. So whenever you will apply for a grant or whatever you are making, you have to, bold you have to write in an energy therapies section, then you have to apply. So history is, in this also, one person, Nielsen Finsen, he got the Nobel Prize. Simple experiment he has done, got through and got the Nobel Prize. So to get a Nobel Prize, or I'll say to do a very wonder, you don't have to do any wonder research. Just only thing is wonder results are required. No need for any complicated research. Simple research, but with best results. Example, low cost. If you do any research with a very low cost you can provide, you'll get a Nobel Prize. Or you will get a Best Researcher Award of India. Indian National Science Academy will give you. Okay, so likewise, so what it can help with, like example, you are in a sunlight, Delhi, summer time, May time, you are going on a road. So what you see, your blood pressure will be high, your everything, you know. Suddenly you wear a black goggles. Your blood pressure, you just measure both the time. So it's a very simple study. Keep yourself in different environment, measure your BP, measure all your inside nutrition, check with the light. It's a new research. Before the medication, with the medication, after the medication, no problem. So 3 level you can do this study. Somebody asked me what is the advantage of this platform of this conference. This is the advantage. We have thousands of ideas. I hope with this, whatever we discussed just now, you must be thinking in your mind, my study, my research, I have done this, okay, I can extend it. This is the benefit of this collaborative platform. So plus in this we have some techniques are there. We can do this study by looking or by exposure of the radiation in your body, 2 ways. Example, this room is there, you remove all the interior, plaster, you are sitting on a very ugly chair, your BP will rise. Make the room very cozy color, BP will be normal. You can do this simple study also. Very simple study. In a room, we do in a pharmacology, the fish we are putting in a different blue color, red color, light color. Why not in the humans? No non-invasive method. Easily you can do any study. Not touching. So wonderful ideas are there. So we have phototherapy, that is utilizing the polychromatic light to promote healing. We have photobiomodulation. Employs, here in this case, we employ the near infrared or EMW, electromagnetic waves, to stimulate the tissue generation and recovery. Physics is, we know light will interact with the object, undergoes reflection, refraction, absorption, all that. That is not our part and parcel. It's not so important. So here we have the different color wheels. Basically 3 colors are there, red, yellow, and blue. When we mix together, the different radiations will come. When we mix further, it will be tertiary. So primary, secondary, and tertiary, 3 levels are there. So I'll not go in much detail. How this, just now I told, you switch on the light and you try to sleep. What you will say? Switch off the light, then only I'll sleep. Because light affects your circadian rhythm, your sleep. Sleep disorder, big research is going in AIMS right now. So light is a biggest, tool for you for research. So in this one example, I'll tell, there it is. I need not go in much detail. Yes, melatonin. A lot of research is going on melatonin right now. So normally at night time, your this concentration is very high. Studies with this melatonin easily we are doing it. So, but with the light you can do. No need to give directly your melatonin, what we are giving, just light exposure. Second example is your serotonin, the opposite effect, daytime hormone. You can do study for that. So likewise, there are, I won't go in much detail. You are very intelligent. We are not computer, but human being have got an idea, which computer doesn't have. Our idea is much more clicking than, yes, in this one more thing is there, AI nowadays. People see, because everywhere AI. AI, the advantage in this is, whatever your prediction of results, prediction of analysis, that will be smoother. Your research will be faster. But one thing I'll tell you, if you want to do your research fast in a short time, have a chemist for in silico study, have a chemist for synthesis characterization, have a pharmacologist for your toxicity evaluation, both cell line at the cellular level and both in vivo, because sometime the animal, the response will not come, the response you have to see on a cellular level. If you are interested, we do cell line studies in AIMS University. So anything, whatever, we can do, cancer, whatever, enzymatic, anything, cellular level, we can do it. So pharmacology, not so high, that is in Sunway, definitely it is there. So anytime you can collaborate with us. Then plus, after that, one formulation scientist we need. In that, you can decide upon, let it be a ayurvedic formulation, let it be a nasal spray, let it be a nasal drop, or whatever, you can plan a formulation scientist. Then you need to have a clinician. If you are going for a orthomolecular medicine, orthomolecularists you need. If you are going for a chromotherapy, chromotherapists you need. Then after that, you need a simple physician. Then if it is a gadget you are using anywhere, you can use a engineer also, you can take a help of it, then make a team, then please apply for a grant, whether nationally or internationally, 100% you will get the grant. But please copyright it. Then apply the fix the certificate, then that's all. So I think that's all from my side. So conclusion is, I need not recite. The conclusion is this only, it's an open field to play. Think about the ideas, see the indication, match with your field, work on 3 levels, your main pharma, your chromotherapy, and your orthomolecular medicine, how you can join together, design some project. If you need any assistance, my number and email ID is in Jamia Hamdard also, with Shahryar sir is there, and with P.R.S. Education Trust also, Dr. Akhil Sharma and Shavita madam is there, so you can ask anytime. So thank you so much. Thanks. Yeah.
Thank you so much, sir, for this very interactive session. Now I would like to announce for the lunch. Everyone, please go ahead for the lunch. And after half an hour, please reassemble here, as we are going to announce our oral and poster winners too. I would like to welcome our valedictory session chief guest, Professor Dr. Vibhu Sahani sir from LLRM Medical College, Meerut. Everyone, please be seated. After lunch, we are going to conclude our scientific lectures of this summit. We are now moved to the valedictory session of this summit. I want to invite our valedictory session chief guest, Professor Dr. Vibhu Sahani, Department of Pharmacy, LLRM Medical College. Sir holds the esteemed position of Superintendent-in-Chief at SVP Patel Hospital, a government hospital with over 20 years of postgraduate experience in teaching, research, and administration. Sir's contribution to the field has been exceptional. His work has led to the publication of numerous articles in highly regarded national and international journals, further cementing his expertise and commitment to advancing pharmaceutical sciences. Sir, please, may we request you to please deliver the valedictory address. And I would like to invite Professor Dr. Mohammad Shahryar sir to felicitate our valedictory session chief guest, Professor Dr. Vibhu Sahani sir. Everyone, please have a big round of applause. Thank you so much, sir. And thank you so much, Professor Dr. Vibhu Sahani sir, to joining us today. Thank you so much, sir. I would like to call upon the stage Professor Dr. Vibhu Sahani sir to deliver the valedictory address. Sir, please.
Thank you very much. Thank you very much, all the dignities present over here. A respected Dr. Vetri Sulam Subramaniam from the Sunway University, Malaysia, Professor Dr. Neera Floria from the IM-HST University, my dear, my lovely friend, Dr. M. Shariar, Dr. Akhil Sharma. He's a backbone of the P.R.S. group of education, Dr. Sweta Sharma, and my dear teachers, colleagues, and all. Thank you very much for giving this opportunity for this particular occasion. And I'm very thankful to all of you that you have got such a patience for me. As the Central Council member of the Pharmacy Council of India, I just want to convey a few messages. Presently, the Pharmacy Council of India is having a lot of litigation, as we all are seeing, facing a lot of challenges also. But one thing I can assure you, that the Pharmacy Council of India will always be with the profession. कुछ चीज़ें past में अभी फार्मेसी काउंसिल ऑफ इंडिया का फ़ायदा उठाइए, उसको यूज़ करिए. कैसे यूज़ करिए? As I am a chairman of the finance committee in the PCI, I have allocated just in the last month only, 30th September sir, 30 सितंबर को अभी मिनिस्ट्री को मैंने अपना बजट द्वारा बना के भेजा, रिवाइज करके, and I allocated a very good handsome amount for the FDP. So my request to all the teachers that you please organize the FDP in their institution with the help of the PCI. We have got a lot of fun. Second, one program is there, a Pharma Anveshan, 6th of March. मैं देखता हूँ, अभी मैं स्क्रूटनी कर रहा था इस साल भी, वही लोग 2-4 यूनिवर्सिटी लास्ट ईयर किया, वही फिर दोबारा अप्लाई कर दिया, वही सुभारती आ गया था और ये सब. तो मेरा कहना ये है कि Subharti is not bad, but every time same university should not. So we should give the opportunity to all the big university, those are functioning in the pharmaceutical education. So my request to you that you should at least apply. आप लोग उसमें apply तो करिए 6 मार्च से पहले कि you will be conducting the Pharma Anveshan in your institute. The Pharma Anveshan, आपको बताऊं, it is a function we are celebrating on the birthday of the father of pharmacy. एम.एल. शराफ साहब के बर्थडे वाले दिन हम इसको ऑर्गेनाइज करते हैं. तो डॉक्टर मंटू कुमार पटेल जी को कन्विंस किया गया था कि हमको एक्चुअली एक रिकग्निशन तो मिले कि व्हाट एग्जैक्टली एम.एल. शराफ सर हैज़ डन फॉर अस. उनका एक विज़न था. अगर आप देखिए 1948 का फार्मेसी एक्ट देखिए, अभी हम फार्माकोपिया मिनिस्ट्री के अंदर फाइल ऑलरेडी हो सकता है कि विंटर सेशन में क्लियर भी हो जाए. इंडियन फार्माकोपिया कमीशन की बात कर रहा हूँ मैं. अगर वो कमीशन बन जाता है तो उसमें मुझे नहीं लगता, आप में से कुछ लोगों ने शायद 2-4-5 लोगों ने देखा हो, पर मेरी बड़ी रिक्वेस्ट रही, हर मंच में जाके मैंने रिक्वेस्ट किया, एक बार आप उस गाइडलाइन को पढ़ो तो सही भाई. 83 पेजेस की गाइडलाइन है, उसको देखो तो सही. मिनिस्ट्री ने आप सबसे सुझाव मांगे थे, पूरे इंडिया के लिए ओपन पोर्टल था, यू गिव योर सजेशन. आपको पता है लास्ट तक कोई सजेशन नहीं आया मिनिस्ट्री के पास। एंड में 2 दिन बाद पहले जब क्लोज हो रहा था डेट, वो फाइल वापस पीसीआई आ गई कि साहब इसमें जो अमेंडमेंट करना है आप कर लो। मुझे अर्जेंट कॉल आया, आपको मेरठ से आना है। मैं आया, मैंने देखा डॉक्टर दीपेंद्र आए रायपुर से, 2-3 लोग और तो जो एकेडमिक्स के थे। तो मैंने सिर पकड़ लिया, उस गाइडलाइन के अंदर हॉस्पिटल फार्मासिस्ट का नाम ही नहीं था। हॉस्पिटल फार्मासिस्ट इज सच ए इम्पॉर्टेंट डिपार्टमेंट, उसका उसमें नाम ही नहीं था। ठीक है, हम भी टीचर्स हैं, आप लोग भी टीचर्स हैं। 90% देयर वाज, डॉक्टर साहब, जो 90% वाज द डिजिग्नेशन गिवन टू द ओनली फार्मेसी टीचर्स। हॉस्पिटल कम्युनिटी फार्मासिस्ट को बिल्कुल साइड कर रखा था, कहीं जिक्र ही नहीं था उनके लिए। तो जब वो हमसे कट जाएंगे तो क्या रह जाएगा। अभी मैं आपको बताऊं, जो हमारा ये टॉपिक है हेल्थ साइंस एंड टेक्नोलॉजी, बड़ा अच्छा टॉपिक रखा डॉक्टर अखिलेश जी ने। अभी मैं नेशनल फार्मा वीक सेलिब्रेशन के फंक्शन में था। थिंक हेल्थ, आई थिंक फार्मासिस्ट वाज द लोगो गिवन बाय द FIP। See, everyone in the world is recognizing the pharmacist. Why not in India? यहाँ पर ही खाली हम लोगों ने डिप्लोमा एसोसिएशन बना रखा है, टीचर एसोसिएशन बना रखा है, इंडस्ट्री एसोसिएशन बना रखा है। माई रिक्वेस्ट, उस दिन भी जाके मैं बोला उस प्रोग्राम में, आई वॉज देयर इन IMA, सो देयर वॉज द प्रोग्राम बाई द ये संयुक्त डिप्लोमा फार्मासिस्ट एसोसिएशन। तो मैंने कहा भाई, वाई इट इज़ डिप्लोमा फार्मेसी एसोसिएशन? B Pharm is also the pharmacy, M Pharm is also pharmacy. Why it is like this? तुम लोग इतना डिफरेंशिएशन क्यों करते हो? डिप्लोमा अलग है, B Pharm अलग है, M Pharm अलग है। Why it is like this? तो मुझे फिर 2 लाइनें वहाँ पे बोलने में मजबूर होना पड़ा, मैं यहाँ भी बोल देता हूँ। फूलों के गुलदस्तों सा महकना हो तो, फूलों के गुलदस्तों सा महकना हो तो साथ जुड़ना होगा। फूलों के गुलदस्तों सा महकना हो तो साथ जुड़ना होगा। यूं तो एक चांद भी है रोशनी के लिए काफ़ी, यूं तो काफ़ी है एक चांद भी रोशनी के लिए, यूं तो काफ़ी है एक चांद भी रोशनी के लिए, गर तारे साथ हों तो मंजर कुछ और ही होगा। तो मैंने कहा तारे कौन है? तो सारे डिप्लोमा फार्मासिस्ट के जो बच्चे थे, वहाँ करीब ये 1,500-2 हज़ार बच्चा था, सब खड़े हो गए कि सर हम तारे। तो मैंने कहा फिर तुम्हारे बिना तो फिर हम अधूरे हो गए ना, यू ऑल आर द एसोसिएटेड पार्ट ऑफ अस ओनली। तो वो लोगों में अभी भी यहाँ पे हमारे, मैं बताऊँ डॉक्टर साहब, हमारे यहाँ पे अभी भी ना डिप्लोमा बच्चों में थोड़ा सा एक इन्फीरिटी कॉम्प्लेक्स रहता है। तो मैं हमेशा उनको अपलिफ्ट करता रहता हूँ कि यू शुड नॉट बी इन्फीरियर, यू आर पार्ट एंड पार्सल ऑफ द फार्मेसी प्रोफेशन। और जो काम कोविड एरा में, कोविड के समय में डिप्लोमा फार्मासिस्ट ने किया, वो काम शायद हम टीचर्स भी नहीं कर पाए। हम भी अपने घरों में बंद थे, है ना, लेकिन वो DM के आदेश पे डिप्लोमा, B Pharm, M Pharm के बच्चों ने जाके, जितने भी प्राइवेट इंस्टीट्यूशन के बच्चे थे, उन्होंने वहाँ वैक्सीनेशन भी किया, बहुत कुछ किया। एनीवे, सो इट इज़ जस्ट द थिंग्स विच आई वांट। सेकंड थिंग, अपने प्यारे बच्चों के लिए, बेटा ये जो प्रोफेशन है ना हमारा, फार्मेसी प्रोफेशन, इसमें ना कभी अपने को अंडर एस्टिमेट मत करना। नेवर थिंक you are some secondary. I am in the medical college. I am sitting with the clinicians and other people. See, I am also a pharmacy person like you. Generally, clinicians have some superiority complex that I'm a consultant. So this problem उनकी inbuilt है, hereditary है, ठीक है? As a pharmacist मैंने भी सोचा कि I will be, भाई I'm also the part and parcel of the medicine, right? So you won't believe, Dr. Cheah, 2016 में the Director General Medical Education has nominated me the Superintendent in Chief of the LLRM Medical College. So 27 departments was under me. Orthopedic, pedia, gastro, ये surgery, medicine, whatsoever, ये gynae सब। तो उनको हजम नहीं हुआ कि एक फार्मेसी का बंदा कैसे, आप में से कोई MBBS तो नहीं है यहाँ पे कोई बात नहीं है तो भी सुन लो। तो फार्मेसी जो है ना, वो एक बरगद के पेड़ के नीचे गुलाब के फूल का खिलना बड़ा मुश्किल होता है ना। तो हम भी इसी तरीके से बेटा संघर्ष करते करते अपने को एस्टेब्लिश किया। तो अगर हम भी सोचते कि हम नॉन क्लिनिकल हैं, नॉन कंसल्ट, मतलब वी आर नॉट ए क्लिनिकल एक्सपर्टाइज, वी आर नॉट हैविंग अ राइट टू प्रिस्क्राइब द मेडिसिन, तो हम लोग भी कहीं हीन भावना में दब जाते। आपको बताऊं, आपको बहुत जल्दी, ऑलमोस्ट फाइनल हो चुका है मिनिस्ट्री के अंदर, रेस्ट्रिक्टेड मेडिसिन में प्रिस्क्रिप्शन राइट मिलने वाले हैं। ठीक है, तो ये मेरे मन में था कि आप लोग अपने आप को सोचते होंगे कि हम लोग दवा नहीं लिख पाते जबकि हम इतना पढ़ते हैं, पर इशू इज़ दिस कि हमको पढ़ना भी पड़ेगा, है ना। हमें क्लासेस में जाना पड़ेगा, लैब में आना पड़ेगा, लैब्स करनी पड़ेंगी। दूसरा, मैं ये जो महसूस कर रहा हूँ, हम ज़्यादा से ज़्यादा, जो टेक्नोलॉजी वर्ड लिखा है, हम ज़्यादा से ज़्यादा एआई पे डिपेंड होते जा रहे हैं। तो मेरी सभी बच्चों से गुजारिश है, रिक्वेस्ट है कि आप लोग एक बहुत ही रेस्पॉन्सिबल प्रोफेशन में हो, आप अपने को उसका गुलाम मत बनाओ, वो आपका गुलाम बने। जितना आप चाहो उतना वो काम करे, आप उस पर डिपेंडेंट मत हो जाओ, क्योंकि अब जो समय की जो डिमांड है, उस डिमांड के अंदर क्रिटिकल थिंकिंग की बहुत आवश्यकता है। अगर आप सोच नहीं पाओगे तो कुछ नया नहीं कर पाओगे, और कुछ नया नहीं कर पाओगे तो आप आउटडेटेड हो जाओगे। ये मेरी 3 बातें आप याद रख लो। ये मैं बच्चों के लिए बोल रहा हूँ, आप सब टीचर्स बैठे हुए हैं, हम लोग अभी इस विषय में, हाँ हाँ, फॉर टीचर्स ऑल्सो, फॉर टीचर्स ऑल्सो। डॉक्टर साहब, मेरा सिलेक्शन ही इसलिए हुआ था, मतलब गॉड गिफ्ट था। आई वॉज इन पब्लिक सर्विस कमीशन, आई वॉज इन इंटरव्यू, तो नीलम वाज माई स्टूडेंट उस समय। तो मेरा डॉक्टर साहब एक्सीडेंट हो गया था, तो आई वॉज ऑन सम स्ट्रिक। तो मुझे लास्ट क्वेश्चन चेयरमैन ऑफ पब्लिक सर्विस कमीशन हैज़ आस्क्ड टू मी, ही कॉल मी 3 टाइम्स। एक और मेरा कंप्यूटर था, वो इक्वल चल रहे थे दोनों, 30 लोग थे, फिर 4 रह गए, फिर 2 रह गए, लास्ट में 2 में से 1 को फाइनल करना था। सो सम चेयरमैन वाज देयर। उस समय ये ब्राइब वगैरह नहीं चलता था कमीशन में, अभी भी पता नहीं चलता है कि नहीं, पर सुनता रहता हूँ। चलो, तो दैट चेयरमैन हैज़ आस्क्ड कि आप तो इतने एक्सपीरियंस हैं, ये सब है, इतने किया हुआ है, बिट्स प्लानी और कॉप्स मनीपाल और ये सब देखा हुआ था पूरा रेज्यूमे, तो बोले आपको तो फिर पढ़ने की कोई ज़रूरत ही नहीं है ना, ऑलरेडी यू हैव स्टडीड लॉट। तो मुझे एक आदत थी, मैं एक बिजनेसमैन था, तो मैं पढ़ाई में ज़्यादा अच्छा नहीं था, राइट। तो मैं नीलम वगैरह की क्लास भी लेके आता था, तो मुझे याद है मार्टिन मेरे हाथ में रहती थी और मैं मार्टिन से थोड़ा बहुत पढ़ के 2-4 पेज आके क्लास लिया करता था। तो मेरी क्योंकि आदत थी कि डेली थोड़ा सा पढ़ के ही क्लास में जाऊँगा, ऐसे ही जाके कुछ भी कर नहीं बोल देने का है। तो मेरे मुंह से निकला कि सर नो, आई हैव टू अपडेट माई सेल्फ एवरी डे। तो उन्होंने शायद इस पॉइंट को पकड़ा, ये तुम बच्चों को तुम्हारे काम आएगा, ठीक है। ओनली ऑन द बेसिस ऑफ दिस लाइन ओनली दे हैव सिलेक्टेड मी, और आज मैं आपके सामने खड़ा हुआ हूँ, राइट। तो जीवन में ना कभी भी कोई भी आदमी कंप्लीट नहीं होता, हर दिन लर्निंग की ज़रूरत है। जिस दिन लर्निंग बंद कर दोगे उस दिन आप स्टेगनेंट हो जाओगे, रुका हुआ पानी बन जाओगे। और उसकी कोई उम्र नहीं होती बेटा। मैं आज भी अपनी बेटी से समझता रहता हूँ, कोई एआई का टूल होता तो मैं उससे कोशिश करता हूँ मुझे बता दे बेटा कैसे करना है। तो लर्निंग की बेटा कोई एज नहीं है। हाँ, थोड़ा स्लो लर्नर हम लोग हो गए हैं, लेकिन कभी उसकी ये मत, यू शुड नॉट फील प्राउड कि मेरे पास काफ़ी नॉलेज हो गई है। यू हैव टू अपडेट योर नॉलेज एवरी डे एवरी टाइम, राइट। और शहर साहब, आपने इतना अच्छा मौका दिया। एक चीज़ बेटा मैं आप लोगों के लिए रेस्पॉन्सिबिलिटी पे 2 लाइनें बोलना चाहूँगा, ध्यान रखना, बहुत ध्यान से सुनना। यू आर इन वेरी रेस्पॉन्सिबल प्रोफेशन, हेल्थ केयर प्रोफेशन। थिंक हेल्थ, think pharmacist. Without pharmacist, health is incomplete. This was the logo from the FIP also. सारे जहाँ की सेहत हमारे कंधों पर है, सारे जहाँ की सेहत हमारे कंधों पर है। ये रहमत खुदा ही है, ये रहमत खुदा ही है कि ये जिम्मेदारी हम बंदों पर है। समझ रहे हैं आप? So we are very lucky, we are very fortunate कि हमको ऊपर वाले ने इस लायक बनाया है। तो बेटा आप लोग कभी भी इसको शॉर्टकट मत निकालना इसका, है ना, कि क्लास क्या करनी, लैपटॉप से पढ़ लेते हैं। नो, गो फिजिकली, इसका मज़ा अलग है। ओके, Thank you very much.
Thank you so much, sir. Before going forward, we want to felicitate our co-chairperson and speakers of this IM-HST Summit 2025. So I would like to call upon the stage Professor Dr. Neeraj Kumar Fuloria, Professor at AIMST University, Malaysia. Sir, please come on the stage. And I would like to call Dr. Shahriar, sir, and Professor Dr. Vibhu Sahani, sir, to felicitate him. Next, I would like to call our co-chairperson and speaker, Professor Dr. Vetri Selvan Subramanian, sir, on the stage. I would like to again call Professor Dr. Vetri Selvan Subramanian, sir, on the stage. Next, I would like to call our resource person, Professor Dr. Rakesh Kumar, sir, Director, Faculty of Pharmacy, BSc College of Engineering and Technology, Mathura. Thank you so much, sir, to joining us today. Next, I would like to call upon the stage our resource person, Dr. Pradeep Kumar, sir, Director, CSS Pharmacy College, Itawa. Thank you so much, sir, to joining us today. And have a great support to us. Thank you so much, sir. Now, moving forward, I want to invite Professor Dr. Shweta Sharma from Galgotias University to present the submission report of the conference. Ma'am, please come on the stage. Everyone have a big round of applause to the ma'am.
Thank you, Akanksha, once again for inviting me here. It was a great day, 2 days here. And I'm going to giving the conclusive remarks on the conference. So the International Conference on Health Science and Technology, IM-HST Summit 2025 was successfully organized by P.R.S. Educational Trust in collaboration with School of Pharmaceutical Education and Research Jamia Hamdard and Sir Jeffrey Cheah Sunway Medical School, Faculty of Medical and Life Science, Sunway University, Malaysia, on 4th and 5th October 2025. So the conference aimed to bring together researchers, academicians, scientists, and industry professionals from across the globe to share their insights, experiences, and latest advancement in health science and technologies. So this event served as a dynamic platform for knowledge exchange, research dissemination, and professional networking. I want to express my gratitude to our chief patron, Professor Dr. Muhammad Afshar Alam Sir, Vice Chancellor, Jamia Hamdard University, and patron, Professor Dr. Raja Effendi, Raja Ali Sir, Dean and Professor, Sir Jaffrey Shee, Sunway Medical School, Faculty of Medical and Life Sciences, Sunway University, for collaborating with us. So, in the starting, the conference commenced with an inaugural ceremony graced by our eminent distinguished, including chief guest, Dr. P.L. Sahu Sir, guest of honor, Dr. Mohan Prasad Sir, patron, Professor Dr. Uma Bhandari Ma'am, and our chairperson, Professor Dr. Muhammad Cheah Sir. The session began with a welcome address by our chairperson, followed by inaugural remark highlighting the importance of global collaboration and innovations in research. The keynote address by our guest of honor, Dr. Mohan Prasad Sir, followed by chief guest Dr. P.R. Sahu Sir, shared their insightful thoughts on health science and technology and importance of dope testing and opportunities in dope testing laboratories. The presidential remark by Dr. Uma Bhandari Ma'am, Dean SPER Jamia Hamdard, provided invaluable insights on emerging trends in health science and technology, setting the tone of the event. As a token of regard, Professor S.K. Singh Memorial Research Award was presented to Professor Dr. Jeanat Iqbal Ma'am, Professor SPER Jamia Hamdard, for her dedicated and remarkable contributions towards research in novel drug delivery system. The Research Excellency Award was presented to Professor Dr. Vetri Selvan Subramaniam Sir, Deputy Dean, Internalization, Department of Biomedical Science, Sir Jaffrey Shee, Sunway Medical School, Faculty of Medical and Life Sciences, Sunway University, Malaysia, for his remarkable contribution in biomedical research. Coming to the technical session, so over the course of the conference, 8 technical sessions were conducted, focusing on emerging trends in science and technology, role of AI in improving the diagnosis and treatment strategies. Distinguished speakers from various institutions presented their findings on cutting-edge topics such as laboratory animals in pharmaceutical research, ethics, technique, and regulations in India, herbal bioenhancers in modern therapeutics, the future of oral care, innovations in medicines and product, and AI-driven technology in silico approaches in drug discovery and drug research, conquering women reproductive cancer through advancement pharmaceutical tools leveraging AI, orthomolecular medicine and chromotherapy, the research avenues, smart biomaterials for targeted anti-obesity drug delivery, current and future approaches in drug discovery and development from natural products. Our eminent speakers were Dr. Vijay Pal Singh Sir, Dr. Sanchit Sharma Sir, Dr. Sanjula Babuta Ma'am, Dr. Professor Jeanat Iqbal Ma'am, Dr. Ozair Alam Sir, Professor Dr. Vetri Selvan Subramaniam Sir, Dr. Mahendran Sekhar Sir, and Professor Dr. Neeraj Kumar Fuloria Sir. Thank you for coming, sir. Now we are approaching to our valedictory session. So it is going today, that is 5th of October 2025, to marking the successful conclusion of the conference. So I welcome our chief guest, Dr. Vibhu Sahni sir, and he lauded the effort of the organizing committee and emphasized the importance of interdisciplinary research and global contributions. So I'm also thankful to our knowledge partners, RV Northland Institute of Technology, IM-HST University, Meerut, and CSS Pharmacy College, Etawah. Now, outcome and recommendations. So, the conferences strengthen international collaboration among participating institutions, provided a global platform for sharing innovative research and best practices, encourage young researchers and scholars to pursue interdisciplinary research. As a word of thanks to Professor Dr. S.K. Abdul Rahman Sir, Dean, School of Medical and Life Sciences, Galgotia University. Thank you, sir. So So, the organizing committee extends heartfelt thanks to all the speakers, delegates, sponsors, and volunteers for their unwavering support. Special thanks to our chairperson, Professor Dr. Mohammad Cheah, Sir, Department of Pharmaceutical Chemistry, SPER, Jamia Hamdard, for providing us encouragement and assistance in marking the conference a grand success. Thank you, sir. So conclusively, this conference completed on a highly successful note and achieving its objective of fostering intellectual exchange and global collaborations. The conference not only facilitated the dissemination of contemporary research, but also inspired future innovations and partnerships. The organizers look forward to hosting similar events in future also to continue promoting academic excellence and research advancement. Thank you to all.
Thank you, ma'am, for submission report of IM-HST Summit 2025. We now come to a special moment, the prize distribution ceremony for the oral and poster presentation. And I think जितने भी presenter हैं वो सभी ही wait कर रहे हैं results का. So I have results. First of all, I am going to announce for the offline presenters, in which we have two categories. First is the oral presentation and second one is the poster. So I would going to announce for the oral presentation Dr. Diksha from Department of Pharmaceuticals, SPER, Jamia Hamdard. She holds the first position in the oral presentation. Everyone have a big round of applause to Dr. Diksha. Soon to be Dr. Diksha. Next, I would like to call upon the stage Dr. Zeenat Iqbal ma'am. Dr. Fawzia ma'am, please come on the stage. Dr. Garima Garg ma'am, please come on the stage. Dr. Shweta ma'am, please come on the stage. Everyone, please have a big round of applause to be soon Dr. Diksha. Next, I would like to call upon the stage our second position holder, Dr. Anzarul Haq from Qatar University. Sir, please come on the stage. Sir is holding the second position in the oral presentation for the offline. Everyone have a big round of applause to the sir. Next, I would like to call upon the stage Dr. Ankita Tiwari from School of Pharmacy, Sarda University. Dr. Ankita Tiwari, Sharda University. Please receive on the behalf of Dr. Ankita Tiwari. अच्छा वो है मैम. Everyone, please give a big round of applause to Dr. Ankita Tiwari from School of Pharmacy, Sharda University. And she holds the third position in oral presentation. Now I am going to announce for the poster presentation first position is held by Muslikuddin Nazumdar from Assam Science and Technology University. He was the first position in the poster presentation. So please give a big round of applause to Muslikuddin Nazumdar. Next, I would like to call upon the stage Swaraj Kumar from NIMS Institute of Pharmacy. And he holds the second position in the poster presentation. Everyone have a big round of applause to Swaraj Kumar from NIMS University. अरे अपने फ्रेंड्स के लिए तालियां बजा दो. Next, I would like to call upon the stage Rishabh Gupta from Sharda University. And he holds the third position in poster presentation. Everyone, please have a big round of applause to Rishabh Gupta. Thank you so much. And now I have result of online presentation. For the oral presentation, the first position goes to Mehak Fatima from Jamia Hamdard. The second position goes to Dr. Pooja Malya, Faculty of Pharmacy, MSR UAS, University, Bangalore. Adiba, third position goes to Adiba Ahmed from Jamia Hamdard. And in third position, we have two members. The second one is Miss Sakshi Nalkande from Srimati Kishori Tai, Bhyor College of Pharmacy, Nagpur, Maharashtra. Next, I have poster presentation online results. And in this, first position is taken by Abdul Feroz Ahmad from NGSM Institute of Pharmaceutical Sciences, NITTE DU. Second position is held by Saurabh Nautiyal from Sardar Bhagwan Singh University, Dehradun. And we have two winners from the second position, and another one is Sandhya Barnawal from United Institute of Pharmacy, Prayagraj. And the last one is the third position, which is held by Kaushalendra Singh from Parul University. Thank you so much, all the delegates to present in our conference and holding these positions. Thank you so much. Now we are moving toward the felicitation of our guests and dignitaries who are really helpful for us and supporting us in this conference very well. So I would like to call, first of all, I would like to call our academic partners, principal, if anyone is here from RV Northland Institute. Anyone from there? IMT University Merit. From there we have Dr. Garima Garg ma'am. Next, I would like to call upon the stage Dr. S.K. Abdul Rahman sir from Galgotias University. Thank you so much, sir, to joining us today. Next, I would like to call Professor Dr. Aftab Alam, sir. Okay. Okay. For the academic partner, I would like to call Professor Dr. Garima Garg, ma'am, from IMT College of Pharmacy, IMT University, Meerut. Next, I would like to call Professor Dr. Aftab Alam, sir, on the stage. Sir is the registration committee chairperson of our conference from Al Karim University, Katihar, Bihar. Thank you so much, sir, for joining us here today. Next, I would like to call upon the stage Dr. Ramji, sir. Next, I would like to call our resource person, Miss Gitika Goyal, ma'am, from Motherhood University, on the stage. Next, I would like to call Mr. Vivek Gautam, sir. From LB Rao Institute of Pharmaceutical Education and Research, Khambat, Anand, Gujarat. Thank you so much, sir, to joining us here. Next, I would like to call Mr. Ayush Agrawal, Indubai Patel College of Pharmacy and Research Center from Gujarat. Everyone, please applaud everyone who is our supporter and our guest here. Thank you so much, sir, to joining us here. Next, I would like to call Dr. Rashid Khan, sir. Okay. Next, I would like to call Professor Dr. Shweta Sharma from Galgotias University, co-chairperson of this summit and a pillar of P.R.S. Educational Trust. Thank you so much, ma'am, for being a great support to us. Thank you so much. Next, I would like to call Dr. Adil Ahmad. From Jamia Hamdard as scientific evaluator. Thank you so much, sir, to joining us. Next, I would like to call upon Dr. Mohammad Rafi Haider, sir, from Jamia Hamdard, who joined us as scientific evaluator. Thank you so much, sir, to joining us. Next, I would like to call Mr. Ashish Verma from P.R.S. Educational Trust on the stage. Next, I would like to call Mr. Ashish Verma on the stage. From P.R.S. Educational Trust. Everyone have a big round of applause to sir. And next, I would like to call Ms. Sunita from P.R.S. Educational Trust on the stage.
Next is Akanksha Sharma, Deputy Director, P.R.S. Educational Trust. May I call Dr. Shekhar Singh on the stage?
I would like to call Dr. Shekhar Singh sir on the stage from BBDIT Lucknow. Okay, thank you so much everyone to joining us. I would like to call upon sir, please wait sir. Sir, please wait. I would like to call upon the stage Dr. Akhil Sharma sir, Chairperson, Director of P.R.S. Educational Trust on the stage. I think this is the first time when we are forcefully felicitate to sir. It is a forceful felicitation, but it should be. Thank you so much, sir, for organizing this beautiful IM-HST Summit 2025. One more announcement is there. I would like to call upon the stage Dr. Kashif Haider, sir from Jamia Hamdard. Thank you so much, sir, to joining us. Everyone have a big round of applause to the sir. I would like to call Mr. Sunil Sharma, sir, from P.R.S. Educational Trust. Thank you so much, sir, to joining us. I would like to call Dr. Wasi Uzzama, Dr. Yaya Sheikh, Mr. Shafiq, Mr. Akram, who have contributed very much in this conference. Without them, we are not able to manage our conference properly. And we felicitate you later, sir. But please come on the stage. Later, we will surely we will felicitate them. Thank you so much, sir, to join us here. And we will surely felicitate you later. And sorry for this inconvenience from P.R.S. Educational Trust. Finally, to extend the heartfelt gratitude on behalf of the organizing committee, I want to invite Professor Dr. Aftab Alam sir on the stage to deliver the vote of thanks.
Hello. Sir, what? Yeah, thanks, Akanksha. Now we are on the climax of the summit, and a vote of thanks. Very good evening to all respected dignitaries, guests, all the delegates, participants in this conference. As we come to close of this remarkable Indo-Malaysian Health Science and Technology Summit 2025, it is my proud privilege to extend a warm vote of thanks to all who made this event a grand success. First and foremost, we express our deep sense of gratitude to our Chief Patron, Professor Dr. Muhammad Afsar Alam, Honorable Vice Chancellor, Jamia Hamdard. We have a big round of applause. For his constant guidance and continued support to this and previous conference organized under the banner of the P.R.S. Educational Trust. Sir, your visionary leadership continues to inspire us to reach higher standards in academics and the research excellence. Our sincere thanks to Professor Dr. Uma Bhandari, Ma'am, Dean SPER Jamia Hamdard, and Professor Dr. Raza Effendi Raza Ali, Dean, Sir Jeffrey Cheah, Sunway Medical School, Malaysia, for their unwavering cooperation and academics partnership in making these Indo-Malaysian collaborations truly meaningful. We extend heartfelt appreciations for our conveners, all conveners, co-conveners, organizing committees, and P.R.S. Educational Trust team members, especially Dr. Akhil Sharma sir, who is the really hero of this summit, we can say. We wish for their dedications, hard work, and meticulous planning that made this summit seamless and impactful. Finally, a big thank you to all our partners, faculty members, students, and volunteers, definitely who put the utmost efforts, hard efforts for these conferences, and their full enthusiasm and their teamwork. With these gratitudes and optimism, we conclude IM-HST 2025. Looking forward to many more such collaborative endeavors in the years ahead. Thank you all and have a wonderful evening. Thanks to everyone. Have a happy journey, happy flight. All the best. Thank you.
With this, we officially conclude the Indo-Malaysian Health Science and Technology Summit 2025. We thank all our dignitaries, speakers, participants, and organizers for making this summit a grand success. And I would like to special thanks to our chairperson, Professor Dr. Mohammad Shahriar sir. Without your guidance, sir, without your guidance, we are not able to complete this, and we are really appreciate your guidance. So thank you so much, sir. Always. Hanji. And also, I would like to thanks our co-chairperson and speaker, Professor Dr. Vetri Selvan Subramanian sir, and Professor Dr. Neeraj Kumar Foloria sir. Thank you so much, sir, to joining us here. Thank you so much. Everyone, please join us for high tea. And offline candidates should get their certificate. Wishing you all the safe travel and continued success in your professional journey. Thank you.